In the quiet moments before dawn, a spider waits motionless in her web while a fly buzzes nearby, unaware of the silk trap that could end its journey. This ancient dance between hunter and hunted has been choreographing the evolution of life for billions of years, shaping not just individual species, but entire ecosystems. From the microscopic pursuit of bacteria to the calculated stalking of apex predators, these relationships have sculpted the architecture of nature itself. Tonight, we explore how predators and prey have written the story of life through their endless, intricate ballet of survival. The relationship between predator and prey begins with the fundamental laws of physics and biology that govern life itself. Before any chase unfolds, before any ambush is set, the basic architecture of living organisms determines who can hunt whom in the grand theater of nature. Size, shape, energy, and the relentless demands of metabolism create the stage upon which all predator prey dramas play out. Consider the simple matter of size. A shrew, weighing barely more than a penny, must consume nearly its own body weight in food each day just to fuel its racing metabolism. Its heart beats over 1,000 times per minute, its body temperature runs hot, and its energy demands are so extreme that it cannot survive more than a few hours without food. This tiny predator lives in a world where every moment is a desperate hunt for the next meal. In contrast, a python may eat a meal equivalent to its entire body weight and then fast for months, its cold-blooded metabolism allowing it to stretch a single, successful hunt across seasons. These metabolic differences create entirely different hunting strategies and lifestyles. The shrew must be constantly active, darting through leaf litter in search of insects, worms, and small prey. Its world is one of perpetual motion and immediate gratification. The python, meanwhile, can afford to wait. It may remain motionless for weeks, conserving energy until the perfect opportunity presents itself for an ambush that will sustain it for months to come. Body size also determines the fundamental physics of movement through different environments. In water, larger predators face less resistance relative to their body size due to the mathematical relationship between surface area and volume. This is why massive great white sharks can cruise efficiently through the oceans, while tiny zooplankton must rely on different strategies entirely. The microscopic world operates under different physical laws, where viscosity dominates over inertia and swimming becomes more like moving through honey than through air. The square cube law governs much of what is possible in predator-prey relationships. As an animal doubles in length, its surface area increases by a factor of 4, but its volume and weight increase by a factor of 8. This means that larger animals have proportionally less surface area for heat loss, making them more efficient at maintaining body temperature, but also more prone to overheating during intense activity. It explains why elephants have large ears for cooling, while shrews burn calories at an almost impossible rate just to stay warm. These physical constraints shape the very architecture of predatory bodies. The streamlined form of a dolphin, the aerodynamic profile of a falcon, the spring-loaded legs of a cat, each represents a solution to the engineering problems posed by the hunt. Speed, after all, is not just about muscle power, but about minimizing drag, maximizing efficiency, and managing the physics of acceleration and deceleration. Flight introduces another dimension of complexity to predator-prey relationships. In the air, the relationship between wing loading, the ratio of body weight to wing area, determines maneuverability versus speed. Sparrowhawks have low wing loading, allowing them to twist and turn through dense forest canopies in pursuit of smaller birds. Peregrine falcons, by contrast, have higher wing loading that enables their spectacular high-speed dives, but makes them less maneuverable at low speeds. These trade-offs mean that different aerial predators excel in different hunting scenarios and pursue different types of prey. The physics of sound also plays a crucial role in predator-prey architecture. Owls have evolved asymmetrical ear openings that allow them to pinpoint the location of prey in three-dimensional space using sound alone. Their flight feathers have specialized structures that eliminate the sound of their own wing beats, making them nearly silent hunters. Meanwhile, their prey species have developed acute hearing to detect the slightest rustle that might signal approaching danger. Energy budgets create some of the most important constraints in predator-prey relationships. Hunting is expensive. A cheetah's explosive sprint may last only seconds before the cat must rest and recover. The enormous energy cost of high-speed pursuit means that cheetahs must be selective about when to commit to a chase. They cannot afford many failed attempts, and this energy constraint shapes everything from their hunting behavior to their social structure. Water presents unique challenges for predators. Sound travels faster and farther underwater than in air, allowing both predators and prey to detect each other at greater distances. However, water is also much denser than air, requiring different adaptations for movement. The torpedo shape, evolved independently in sharks, dolphins, and tuna, represents a convergent solution to the problem of moving efficiently through a dense medium. Temperature adds another layer of complexity to predator-prey architecture. Cold-blooded predators, like snakes and lizards, depend on external heat sources to maintain the body temperatures necessary for effective hunting. A rattlesnake in cool weather moves slowly and strikes with less force than the same snake in warm conditions. This temperature dependence creates windows of vulnerability and opportunity that warm-blooded prey species can exploit. The sensory systems of predators and prey represent some of the most remarkable examples of biological engineering. The compound eyes of dragonflies process visual information fast enough to track prey moving at high speeds, while their four wings can beat independently, allowing them to hover, fly backward, and execute sharp turns that would challenge any human-built aircraft. Their prey, smaller flying insects, have evolved their own evasive strategies, including erratic flight patterns that make their movements unpredictable. Some predators have pushed sensory capabilities to extraordinary extremes. Sharks can detect electrical fields as weak as 5 billionths of a volt, allowing them to sense the muscle contractions of hidden prey. Snakes in the pit viper family can detect temperature differences as small as 0.003 degrees Celsius, enabling them to hunt warm-blooded prey in complete darkness. Bats have refined echolocation to such a degree that they can distinguish between objects differing in thickness by mere fractions of a millimeter. The architecture of teeth, claws, and other weapons reveals the intimate connection between form and function in predator design. The serrated teeth of sharks are designed to slice through flesh and create wounds that weaken prey through blood loss. The canine teeth of cats are precisely spaced to slip between the vertebrae of their prey, severing the spinal cord with surgical precision. Each design represents millions of years of refinement in the art of killing. Consider the engineering marvel of a spider's web. Different species construct webs with varying architectures depending on their hunting strategies. Orb weavers create circular webs that intercept flying insects, while funnel web spiders build sheet webs with retreat tunnels that allow for quick ambush attacks. The silk itself varies in properties. Some strands are sticky to trap prey, others are strong to anchor the web, and still others are designed to transmit vibrations that alert the spider to captured prey. Defensive architecture is equally sophisticated. The armored shells of turtles, the quills of porcupines, and the toxic spines of lionfish all represent different solutions to the problem of avoiding becoming someone else's meal. Some defenses are passive, like the hard shells that make prey difficult to consume. Others are active, like the ability of many lizards to shed their tails when grasped by a predator, or the spray of noxious chemicals that bombardier beetles can direct at attackers with remarkable accuracy. The microscopic world reveals predator-prey relationships operating under entirely different physical principles. Here, molecular forces dominate, and predatory bacteria must navigate a world where Brownian motion constantly buffets them about. Some have evolved rotating flagella that work like tiny propellers, while others use chemical gradients to navigate toward their prey. At this scale, the Reynolds number, a measure of fluid dynamics, is so low that stopping and starting require different strategies than those used by larger organisms. Plant architecture, too, has been shaped by the pressure of herbivorous predators. Thorns, toxic compounds, and thick bark all represent defenses against creatures that would consume plant tissues. Some plants have evolved to be unpalatable or even deadly to herbivores, turning the tables on would-be predators. The acacia tree's relationship with ants demonstrates how plants can recruit bodyguards, providing shelter and food in exchange for protection from herbivorous insects. The energy economics of predation create fundamental limits on what is possible in nature. Large predators need large prey to make their hunts energetically worthwhile, while small predators must content themselves with smaller meals but can afford to be less selective. This size-based partitioning helps explain why ecosystems can support multiple predator species without complete competitive exclusion. A lion and a shrew may both be predators, but they operate in entirely different energetic worlds. Biomechanics also plays a crucial role in determining success. The strike of a mantis shrimp is one of the fastest movements in the animal kingdom, accelerating faster than a bullet and generating forces capable of breaking through the shells of crabs and snails. The power comes from a spring-loaded mechanism in the shrimp's arms that stores energy like a compressed bow before releasing it in an explosive strike. As we consider these fundamental constraints, we begin to see how the basic physics and biology of life create the stage for more complex interactions. The architectural solutions that have evolved to meet these challenges set the foundation for the evolutionary arms races that have driven much of the diversity we see in nature today. Each physical adaptation, each metabolic strategy, each sensory capability represents a move in an ancient game where the stakes are survival itself and the rules are written in the unchanging laws of physics and chemistry that govern all life on Earth. These constraints create the boundaries within which evolution operates, but within those boundaries, life has found an almost infinite variety of solutions to the challenges of hunting and avoiding being hunted. The fundamental constraints of physics and biology create the arena, but evolution writes the script. In the endless theater of predator and prey, each successful adaptation by one species creates pressure for counter-adaptations in the other, launching evolutionary arms races that have been running for hundreds of millions of years. These races have no finish line, no ultimate winner, only an endless spiral of innovation and counter-innovation that has driven much of the spectacular diversity we see in the natural world today. Consider the relationship between cheetahs and their prey on the African savanna. The cheetah's incredible speed, capable of reaching 70 miles per hour in short bursts, represents the culmination of millions of years of evolutionary refinement. Every aspect of the cheetah's anatomy has been honed for speed: enlarged heart and lungs to pump oxygen to muscles, flexible spine that acts like a spring, lightweight build that sacrifices power for acceleration, and even semi-retractable claws that provide traction like running spikes. Yet their primary prey, gazelles and impalas, have not simply accepted defeat in this race for speed. They have evolved their own counter strategies. While they cannot match the cheetah's top speed, they excel in endurance and maneuverability. A gazelle can maintain moderate speeds much longer than a cheetah can sustain its sprint, and their ability to make sharp turns at high speed often allows them to escape even when caught by surprise. The cheetah's lightweight build, so crucial for speed, becomes a liability in the wrestling match that ensues if the initial takedown fails. This arms race has pushed both species to extremes. Cheetahs have become so specialized for speed that they have sacrificed much else. They are relatively weak compared to other big cats, cannot climb trees effectively, and are easily displaced from their kills by stronger predators. Gazelles, meanwhile, have developed early warning systems through enhanced vision and hearing, and employ group vigilance strategies where multiple pairs of eyes watch for danger while others feed. The evolutionary pressure is mutual and relentless. A slightly faster cheetah has better hunting success and leaves more offspring. A slightly more agile gazelle survives more encounters and lives to reproduce. Over thousands of generations, these small advantages compound, driving both species toward ever greater extremes of performance. Neither can afford to rest on evolutionary laurels. The other species is always evolving too. Bats and moths provide perhaps the most dramatic example of an escalating sensory arms race. When bats first evolved echolocation some 50 million years ago, they gained a tremendous advantage in hunting night-flying insects. Their high-frequency calls bounce off objects in the environment, creating detailed sonic maps that allow them to navigate and hunt in complete darkness. This biological sonar is so sophisticated that bats can distinguish between different types of insects, avoid obstacles as thin as wires, and even detect insects hiding motionless on leaves. Moths, however, did not passively accept their role as easy prey. Many species evolved ears sensitive to ultrasonic frequencies, allowing them to detect approaching bats and take evasive action. Some moths can hear bat calls from distances of over 100 feet, giving them precious seconds to dive, turn, or execute erratic escape maneuvers that make them difficult targets. But the arms race was far from over. Some bats responded by evolving stealth echolocation, calls so quiet that moths cannot detect them until the bat is dangerously close. Others began using frequencies outside the hearing range of their prey, essentially finding new acoustic channels for their sonar. Still others evolved to hunt using passive listening, detecting the sounds made by insects themselves rather than relying entirely on echolocation. The moths struck back with even more sophisticated defenses. Some species evolved the ability to jam bat sonar by producing their own ultrasonic clicks, essentially creating acoustic camouflage. Others developed sound-absorbing scales on their wings that reduced the echoes returned to hunting bats. Tiger moths took the defensive arms race to its extreme by evolving toxic compounds and warning sounds that teach bats to avoid them entirely. This sensory arms race continues today, with new adaptations appearing in both bat and moth populations. Scientists have documented cases where moth populations show rapid evolutionary responses to bat predation pressure within just a few decades, demonstrating that these arms races operate on timescales we can observe directly. Venomous snakes and their prey represent another fascinating arms race, but one fought with chemistry rather than speed or senses. Snake venoms are cocktails of dozens or even hundreds of different proteins and enzymes, each targeting different physiological systems in prey animals. Some components attack the nervous system, causing paralysis. Others break down blood vessels and tissue. Still others prevent blood clotting, causing internal bleeding. But prey species have not been defenseless in this chemical warfare. Many have evolved resistance to the venoms of snakes in their environment. Ground squirrels in areas with rattlesnakes show remarkable resistance to rattlesnake venom, surviving bites that would kill squirrels from areas without venomous snakes. This resistance comes at a cost. The biochemical changes needed to neutralize venom require energy and may compromise other physiological functions. The snakes, in turn, have evolved more potent venoms. Rattlesnakes in areas with resistant ground squirrel populations have more toxic venom than those in areas where prey lacks resistance. This creates a geographic mosaic of different venom potencies and resistance levels, with each population locked in its own local arms race. Some prey species have taken the chemical arms race in unexpected directions. Certain populations of garter snakes have evolved resistance to the tetrodotoxin in their newt prey, allowing them to eat newts that would be deadly to other predators. The newts, meanwhile, have responded by evolving even higher concentrations of toxin. In some areas, newts carry enough tetrodotoxin to kill dozens of adult humans, yet local garter snakes can consume them safely. The arms race between predatory fish and their prey has driven innovations in body armor and weaponry. Many fish species have evolved protective scales, spines, or bony plates that make them difficult to swallow. Pufferfish can inflate themselves to several times their normal size, becoming too large for most predators to consume. Surgeonfish have evolved razor-sharp spines near their tails that can inflict serious wounds on attackers. Predatory fish have responded with their own adaptations. Some have evolved pharyngeal jaws, a second set of jaws in their throats that can process heavily armored prey. Others have developed crushing dentition capable of breaking through shells and armor. Moray eels have taken this to an extreme, with pharyngeal jaws that can actually extend forward to grasp prey and pull it back into the throat. The evolutionary arms race extends beyond individual encounters to include deception and information warfare. Many predators have evolved camouflage that makes them nearly invisible to prey. The leafy sea dragon looks so much like floating seaweed that small fish and crustaceans may not recognize the danger until it's too late. Crab spiders can change color to match the flowers they hunt on, becoming deadly decorations that ambush visiting insects. Prey species have developed their own deceptive strategies. Many have evolved eye spots, large conspicuous markings that look like the eyes of a much larger animal. When threatened, they can suddenly reveal these markings, potentially startling predators long enough to escape. Butterfly fish have eye spots near their tails that may confuse predators about which end is the head, causing attacks to miss vital organs. Mimicry represents perhaps the most sophisticated form of evolutionary deception. Harmless species evolve to look like dangerous ones, gaining protection without having to invest in actual defenses. The scarlet king snake mimics the coral snake's warning coloration so precisely that even experienced herpetologists sometimes need to examine them closely to tell them apart. This mimicry only works because coral snakes are genuinely dangerous. The king snake is essentially borrowing the coral snake's reputation for deadliness. Some species have taken mimicry to extraordinary lengths. Certain spiders have evolved to look remarkably like ants, gaining protection from predators that avoid the aggressive, well-defended social insects. These spider mimics don't just look like ants, they move like them too, waving their front legs to mimic antennae and adopting the characteristic zigzag walking pattern of ants. The arms race between predators and prey has also driven the evolution of increasingly sophisticated behaviors. Many prey species have developed complex alarm systems that allow them to communicate about different types of threats. Vervet monkeys have different calls for different predators, one for leopards, another for eagles, and a third for snakes. Each call triggers appropriate escape responses. The leopard call sends monkeys up into trees. The eagle call makes them look up and seek cover, and the snake call causes them to stand on their hind legs and scan the ground. Some predators have learned to exploit these communication systems. Certain hawks have been observed hiding near areas where small birds commonly give alarm calls, essentially eavesdropping on their prey's communication network. Other predators use deceptive signals, like the alligator snapping turtle that uses a worm-like appendage in its mouth to lure fish within striking range. Tool use has entered the arms race as well. Sea otters use rocks to smash open shellfish, while some birds drop mollusks from height onto rocks to break them open. Egyptian vultures throw rocks at ostrich eggs to crack them. On the defensive side, some primates have learned to use sticks to extract insects from crevices where predators might be hiding, and dolphins sometimes use marine sponges to protect their sensitive rostrums while foraging in areas with dangerous coral formations. The temporal dimension adds another layer to evolutionary arms races. Many predators and prey have evolved different activity patterns to avoid each other. Diurnal predators hunt during the day, while their prey may become nocturnal. But this creates opportunities for nocturnal predators, which may then drive some prey species to become active during dawn and dusk, when neither day nor night specialists are most effective. These arms races have no end point because the environment itself is constantly changing. Climate change, habitat modification, and the introduction of new species continually alter the rules of engagement. What works today may be obsolete tomorrow, and evolution must constantly respond to new challenges and opportunities. Perhaps most remarkably, these arms races have become self-accelerating. As predators and prey become more sophisticated, the evolutionary pressure on both sides intensifies. A more effective predator creates stronger selection pressure for better defenses, which in turn creates pressure for even more effective predation strategies. This positive feedback loop has driven much of the complexity and diversity we see in modern ecosystems. The weapons and defenses produced by these evolutionary arms races form the foundation for the next chapter in our exploration, the remarkable arsenal of chemical weapons that predators and prey have developed in their ongoing struggle for survival. In the microscopic realm, where molecules dance and reactions unfold in milliseconds, predators and prey wage war with weapons more sophisticated than any human arsenal. Chemistry has become both sword and shield in nature's endless battle, creating compounds so precisely engineered that they can paralyze a victim in seconds or protect a defender for a lifetime. This molecular battlefield has produced some of the most elegant and terrifying instruments of death and survival ever devised. The cone snail, a seemingly innocent mollusk found in tropical waters, carries within its shell one of nature's most sophisticated chemical weapons laboratories. When hunting, it extends a harpoon-like radula loaded with conotoxins, peptides so specifically designed that different varieties target different ion channels in nerve cells with surgical precision. Some conotoxins block sodium channels, preventing nerve impulses from traveling. Others target calcium channels, disrupting the release of neurotransmitters. Still others affect potassium channels, altering the electrical properties of nerve membranes. What makes these toxins particularly remarkable is their specificity. Each conotoxin is like a molecular key designed to fit one particular lock in the nervous system. The geography cone snail produces over 100 different conotoxins, creating a chemical arsenal that can be fine-tuned for different prey species. When hunting fish, it deploys fast-acting toxins that cause immediate paralysis. When targeting worms, it uses different compounds that work more slowly but are equally effective. The blue-ringed octopus takes chemical warfare to another level entirely. This small cephalopod, no larger than a human hand, carries enough tetrodotoxin to kill 26 adult humans within minutes. The toxin works by blocking sodium channels in nerve cells, preventing the transmission of electrical signals. Victims remain conscious but cannot move or breathe, experiencing what researchers describe as a fully aware paralysis. The octopus manufactures this compound in specialized glands and can inject it through its bite, turning feeding into a form of chemical assassination. But perhaps no predator has mastered the art of venom like snakes. Snake venoms are not single compounds, but complex cocktails of proteins, enzymes, and peptides that work together to immobilize and digest prey. A single bite from a gaboon viper delivers a mixture of hemotoxins that destroy red blood cells, cytotoxins that break down tissue, neurotoxins that paralyze the nervous system, and anticoagulants that prevent blood clotting. The sophisticated chemistry doesn't stop there. Many snake venoms contain hyaluronidase, an enzyme that breaks down connective tissue and acts as a spreading factor, helping other toxins penetrate deeper into the victim's body. Phospholipase, two enzymes destroy cell membranes, while metalloproteinases break down the extracellular matrix that holds tissues together. The result is a biological weapon system that simultaneously attacks multiple physiological targets. Different snake species have evolved venoms tailored to their preferred prey. Sea snakes that hunt fish have developed highly potent neurotoxins that cause rapid paralysis, preventing prey from swimming away. Vipers that hunt mammals rely more heavily on hemotoxins and tissue-destroying enzymes that cause internal bleeding and begin the digestive process while the prey is still alive. Elapids like cobras and mambas combine both strategies, using neurotoxins for rapid immobilization and cytotoxins for tissue destruction. The evolutionary refinement of these chemical weapons is ongoing. Researchers have discovered that snake venoms can vary not just between species, but between populations of the same species. Rattlesnakes in areas with ground squirrels that have evolved venom resistance have responded by developing more potent toxins. This geographic variation in venom composition creates a mosaic of local arms races, with each population fine-tuning its chemical arsenal to match local prey defenses. Spiders have developed their own approach to chemical predation. Unlike snakes, which inject large volumes of venom, spiders typically deliver smaller, more concentrated doses of highly specific toxins. The black widow's latrotoxin targets the nervous system by causing massive release of neurotransmitters, essentially overwhelming the victim's neural circuits. Funnel-web spider venom contains atracotoxins that keep sodium channels permanently open, causing continuous nerve firing and rapid death. Some spiders have evolved venoms so precisely calibrated that they can adjust the dose based on prey size. The prey becomes paralyzed but remains alive, allowing the spider to feed on fresh tissue over extended periods. This biological precision dosing ensures that the prey doesn't die immediately and spoil, while also preventing any possibility of escape. Cnidarians, jellyfish, sea anemones, and their relatives represent perhaps the most ancient form of chemical warfare in the animal kingdom. Their nematocysts are microscopic harpoons that fire with such force they can penetrate human skin and inject venom in less than a millisecond. The box jellyfish's venom contains toxins that attack the cardiovascular system, nervous system, and skin simultaneously, causing excruciating pain and potentially fatal cardiac arrest. Each nematocyst is a marvel of bioengineering, containing a coiled thread under tremendous pressure and the marvel of bioengineering 8. When triggered by chemical or mechanical stimuli, the thread fires out with an acceleration of over 5 million g-forces, among the fastest biological processes known to science. The thread carries toxins specifically designed to penetrate cell membranes and disrupt cellular function. Plants, though unable to flee from herbivorous predators, have developed some of the most sophisticated chemical defenses in nature. Alkaloids like nicotine, caffeine, and morphine are plant defense compounds that interfere with animal nervous systems. Nicotine, found in tobacco plants, binds to acetylcholine receptors and can cause paralysis in insects at concentrations that would be lethal to most small animals. The castor bean plant produces ricin, a toxin so potent that just a few molecules can kill a cell by shutting down protein synthesis. Foxglove contains digitalin and other cardiac glycosides that interfere with heart function, causing irregular heartbeat and potentially fatal arrhythmias. These compounds are so precisely engineered that many have found use in human medicine. Digitalis from foxglove is still used to treat certain heart conditions, while morphine from poppies remains one of our most effective painkillers. Some plants have evolved chemical defenses that specifically target the digestive systems of herbivores. Tannins bind to proteins and make plant tissues difficult to digest, essentially creating biological leather in the mouths of would-be grazers. Protease inhibitors interfere with digestive enzymes, preventing herbivores from extracting nutrients from plant tissues. These compounds can reduce the nutritional value of plant material so dramatically that herbivores must find alternative food sources or face malnutrition. The defensive use of chemistry extends to chemical communication and warning systems. Many animals that have evolved potent chemical defenses advertise this fact through bright warning colors and distinctive odors. Poison dart frogs, despite their small size, carry alkaloids so toxic that indigenous peoples have used them to poison arrow tips for centuries. The frog's brilliant colors serve as warnings to potential predators that eating them would be a fatal mistake. Skunks represent a different approach to chemical defense, the use of noxious compounds to deter rather than kill attackers. Skunk spray contains thiols, sulfur-containing compounds that create an overwhelmingly unpleasant odor that can be detected by human noses at concentrations as low as a few parts per billion. The spray also causes temporary blindness and respiratory irritation, giving the skunk time to escape while its attacker recovers. Bombardier beetles have evolved perhaps the most dramatic chemical defense system in the insect world. When threatened, they can explosively spray a boiling chemical mixture that reaches temperatures of over 100 degrees Celsius. This biological cannon works through a two-chambered system where hydrogen peroxide and hydroquinones are mixed with catalase and peroxidase enzymes, creating an instantaneous chemical reaction that produces the scalding defensive spray. The chemistry of defense extends to more subtle molecular strategies. Many prey species have evolved the ability to detect and neutralize toxins before they can cause damage. Liver enzymes in many mammals can metabolize plant alkaloids, rendering them harmless. Some animals have evolved modified versions of target proteins that are less susceptible to particular toxins, essentially creating molecular armor against chemical attack. The arms race between toxic predators and resistant prey has driven remarkable innovations on both sides. Monarch butterflies sequester toxic compounds from milkweed plants in their bodies, making them unpalatable to birds. But some birds have evolved the ability to detect and avoid the most toxic parts of the butterfly while eating the less dangerous tissues. This has led to an ongoing evolutionary dance where butterflies must balance toxicity levels. Too little, and they remain vulnerable. Too much, and they poison themselves. Venomous animals face the constant challenge of evolving resistance to their own toxins. Snakes must be immune to their own venom, requiring the evolution of modified target proteins or enhanced detoxification systems. This self-protection often comes at a metabolic cost, requiring energy that could otherwise be used for growth or reproduction. The chemical battlefield also includes pheromones and other signaling molecules that can manipulate behavior rather than directly causing harm. Some predatory insects release compounds that mimic the sex pheromones of their prey, luring victims into range. Certain parasites can manipulate host behavior through neurochemical compounds, effectively hijacking the host's nervous system to serve the parasite's reproductive needs. Recent research has revealed that the chemical arms race extends to the molecular level within cells themselves. Some bacterial predators inject toxins that specifically target the cellular machinery of their prey, while the prey bacteria evolve resistance mechanisms and even toxin-degrading enzymes. This microscopic chemical warfare plays out in environments from soil to the human gut, influencing everything from nutrient cycling to human health. The sophistication of these chemical weapon systems continues to inspire human technology. Researchers study cone snail toxins to develop new painkillers that target specific nerve pathways without the side effects of traditional opioids. Spider venoms are being investigated as potential sources of insecticides that are toxic to pests but harmless to beneficial insects. The precision of these biological systems often surpasses anything we can engineer in laboratories. As we delve deeper into the molecular mechanisms of these chemical defenses, we begin to see how they connect to broader patterns of survival strategy. The investment in chemical weapons represents just one approach to the fundamental challenge of avoiding detection and capture. In our next exploration, we turn to the visual realm, where predators and prey have developed equally sophisticated strategies of concealment, deception, and disguise. In the theater of survival, visibility often means death. While chemical warfare operates in the realm of molecules, the visual battlefield spans the entire spectrum of light, from ultraviolet frequencies, invisible to human eyes, to the deep infrareds that reveal heat signatures in the darkness. Here, predators and prey have engaged in an artistic arms race, developing techniques of concealment, deception, and illusion that would challenge the greatest masters of camouflage and stagecraft. The art of invisibility begins with understanding light itself. Most animals see the world through eyes adapted to detect specific wavelengths of electromagnetic radiation, typically those that penetrate water or atmosphere most effectively. But this creates opportunities for those clever enough to exploit the blind spots and limitations of visual systems. The transparency of glass frogs represents one of nature's most elegant solutions to the problem of visibility. Their skin, organs, and even blood become virtually invisible, allowing light to pass through their bodies with minimal distortion. This transparency is no simple matter of evolution removing pigments. The glass frog's tissues have been precisely engineered at the cellular level to minimize light scattering. Specialized cells called iridophores contain crystalline platelets that bend light in specific ways, while the arrangement of muscle fibers and blood vessels has been optimized to reduce shadows and reflections. The result is a creature that can rest on leaves in broad daylight while remaining nearly invisible to predators scanning from below. Cuttlefish represent perhaps the pinnacle of active camouflage technology. These cephalopods possess millions of chromatophores, specialized cells containing pigment granules that can be expanded or contracted by surrounding muscle fibers. Beneath these lie iridophores filled with reflective proteins that create metallic sheens and shifting colors. Deeper still, leucophores scatter light to create white and silver tones. The entire system operates under neural control, so sophisticated that a cuttlefish can display different patterns on different parts of its body simultaneously. The speed of these color changes defies belief. A cuttlefish can shift from sandy brown to mottled green to striped silver in fractions of a second, continuously adjusting its appearance to match its surroundings. But this is not merely reactive camouflage. Cuttlefish can anticipate the backgrounds they're moving toward and begin changing color before they arrive. They can even create false textures, raising papillae on their skin to mimic the three-dimensional structure of coral, rocks, or seaweed. Chameleons employ a different but equally sophisticated approach to active camouflage. Their color changes result from the arrangement of nanocrystals within specialized cells called iridophores. By altering the spacing between these crystals, chameleons can tune their reflectance to different wavelengths of light. When the crystals are closely packed, they reflect blue light. When spread apart, they shift toward red. This gives chameleons precise control over their coloration, allowing them to blend with backgrounds while also communicating emotional states and territorial claims. Static camouflage, though less dramatic than active color change, can be equally effective. The bark mantis has evolved such precise mimicry of tree bark that it includes fake lichen patches, simulated insect damage, and even artificial moss growths. Every detail of its body's surface tells the story of a weathered piece of wood, complete with the random imperfections that make the disguise convincing to both predators and prey. Stick insects take structural mimicry to extraordinary lengths. Not only do they resemble twigs in color and texture, but they have evolved body proportions, joint angles, and even swaying behaviors that perfectly mimic branches moving in the wind. Some species go further, developing wing patterns that look like bite marks or disease spots on leaves, adding layers of detail that make their disguises nearly perfect. The peppered moth provides one of the most famous examples of camouflage adaptation in action. Before industrial pollution darkened tree trunks with soot, light-colored moths blended perfectly with lichen-covered bark. As pollution increased, dark-colored variants gained an advantage on the newly blackened trees. When pollution controls cleaned the air, light-colored forms again became more common. This rapid evolutionary response demonstrates how quickly camouflage patterns can adapt to changing environments. Counter-illumination represents one of the most ingenious approaches to camouflage ever evolved. Many deep-sea creatures produce light on their undersides to match the faint illumination filtering down from above, effectively erasing their silhouettes when viewed from below. The cookie-cutter shark and many species of squid have perfected this technique, using bioluminescent photophores arranged in precise patterns to create the optical illusion of empty water. Some animals have taken counter-illumination a step further, developing dynamic lighting systems that adjust to changing conditions. Certain species of deep-sea fish can modulate the intensity and color of their bioluminescence to match the changing light levels as they move through different depths. This requires not only the ability to produce light, but also sophisticated optical sensors to measure ambient illumination and neural systems to coordinate the response. Disruptive coloration breaks up the outline of an animal's body, making it difficult for predators or prey to recognize familiar shapes. Zebras' stripes may serve this purpose, creating optical illusions that confuse predators about the size, speed, and direction of movement of individual animals within a herd. The high contrast patterns can cause a visual effect called motion dazzle, where rapid movement creates strobing patterns that make it difficult to track a target. Many fish species employ similar principles with bold stripes or spots that break up their body outlines. Angelfish have eye stripes that conceal their actual eyes, while fake eye spots near their tails may confuse predators about which direction they're facing. This head-tail confusion can cause attacks to miss vital organs, giving the fish a chance to escape. Eye spots represent one of the most psychologically sophisticated forms of visual deception. Large, conspicuous markings that resemble the eyes of much larger animals can trigger instinctive fear responses in potential predators. When threatened, many moths and butterflies suddenly reveal hidden eye spots on their hindwings, creating the illusion that a small, harmless insect has transformed into a large, dangerous predator. The startle effect often provides just enough time for escape. Some species have evolved eye spots so realistic they include highlights, shadows, and even apparent pupils. The peacock mantis shrimp's threat display reveals eye spots so convincing that they can intimidate predators many times larger than the mantis shrimp itself. These false eyes exploit deep-seated evolutionary programming that causes most animals to be wary of direct eye contact from potential threats. Aggressive mimicry flips the usual predator-prey dynamic by having predators disguise themselves as harmless or even beneficial organisms. Orchid mantises have evolved to look remarkably like flower petals, complete with the delicate colors and textures that attract pollinating insects. They position themselves within real flower clusters and wait motionlessly for prey to approach what appears to be just another bloom. The alligator snapping turtle employs perhaps the most famous example of aggressive mimicry in freshwater environments. A specialized appendage on its tongue looks remarkably like a small worm, complete with realistic movements that mimic a struggling invertebrate. Fish approaching what they believe to be an easy meal find themselves facing crushing jaws capable of severing fingers. Some predators mimic not just the appearance, but also the behavior of their models. Zone-tailed hawks have evolved coloration and flight patterns nearly identical to turkey vultures, allowing them to soar among groups of scavengers without alarming potential prey below. Small animals that have learned to ignore the harmless vultures overhead may not recognize the danger until the disguised hawk strikes. Batesian mimicry allows harmless species to gain protection by resembling dangerous ones. The viceroy butterfly's orange and black coloration closely resembles that of the toxic monarch butterfly, gaining protection from predators that have learned to avoid the unpalatable model. This form of mimicry only works when the mimic is less common than the model. If harmless mimics become too numerous, predators learn that the warning signals are unreliable. Mullerian mimicry involves multiple dangerous species converging on similar warning patterns, reinforcing the message that certain color combinations signal danger. Various species of wasps, bees, and toxic beetles share similar yellow and black warning patterns, creating a network of mutual protection. Each predator's bad experience with any member of the group reinforces avoidance of the entire complex. Some species have evolved sophisticated mimicry complexes involving multiple models and forms. Heliconius butterflies in South America participate in large mimicry rings, where dozens of species share similar wing patterns. Within each geographic region, local species converge on area-specific patterns, creating a mosaic of different mimicry complexes across the continent. The precision required for effective mimicry extends beyond simple coloration to include details of size, shape, movement, and even timing. Ant mimicking spiders must not only look like ants, but also move like them, adopting the characteristic zigzag gait and using their front legs to mimic antennae. They must also be active during the same times of day as their ant models and often restrict themselves to areas where ants are commonly found. Seasonal changes add another layer of complexity to visual deception strategies. Arctic animals like ptarmigan and Arctic foxes change from brown summer coats to white winter plumage or fur, maintaining their camouflage effectiveness as snow cover changes their environment. These seasonal changes require precise timing. Changing too early or too late can leave an animal conspicuous against the wrong background. Some predators have learned to exploit the camouflage strategies of their prey. Certain birds have developed search images that help them detect camouflaged insects, learning to recognize the subtle cues that betray hidden prey. This has led to an ongoing evolutionary cycle where camouflage becomes more sophisticated in response to increasingly discriminating predators. The underwater environment presents unique challenges for visual deception. Water filters out different wavelengths of light at different depths, meaning that red camouflage, effective in shallow water, becomes meaningless in deeper areas where red light cannot penetrate. Many deep-sea creatures are predominantly red or black, colors that appear identical in the blue-filtered light of the deep ocean. Bioluminescence adds another dimension to underwater camouflage and communication. Many marine creatures use patterns of light-producing organs not just for counter-illumination, but also for species recognition, territorial displays, and predator confusion. Some species can create complex light shows that may serve to disorient predators or coordinate group behaviors. The effectiveness of any camouflage strategy depends not only on the visual system of the intended audience, but also on the environmental context. Patterns that provide perfect concealment in one habitat may be conspicuous in another. This has led to the evolution of behavioral adaptations that ensure animals position themselves in appropriate backgrounds. Many cryptic species show strong preferences for resting sites that complement their camouflage patterns. Advanced camouflage systems often integrate multiple sensory modalities beyond just vision. Some animals coordinate their visual camouflage with chemical signals, acoustic properties, or even electromagnetic signatures to create comprehensive concealment systems. The most successful masters of disguise understand that true invisibility requires attention to every detail that might betray their presence. As we observe these remarkable adaptations for concealment and deception, we begin to see how they connect to broader survival strategies that extend beyond individual encounters. The evolution of sophisticated visual deception has paved the way for complex social behaviors and group strategies that represent the next frontier in predator-prey relationships. The transition from solitary to social living represents one of the most profound transformations in the history of predator-prey relationships. When individual hunters began cooperating and isolated prey started forming groups, the fundamental rules of engagement shifted dramatically. This social revolution has created some of nature's most sophisticated hunting strategies and elaborate defense systems, fundamentally altering the balance of power in ecosystems around the world. Pack hunting transforms predators from individual opportunists into coordinated killing machines capable of bringing down prey many times larger than any single hunter could manage alone. Wolves exemplify this transformation. A lone wolf typically weighs between 60 and 100 pounds, yet a coordinated pack can successfully hunt moose weighing over 1,000 pounds. This dramatic scaling of hunting capability emerges not from individual strength, but from the complex social dynamics that allow multiple predators to function as a single coordinated unit. The sophistication of wolf pack hunting strategies rivals military tactics. Scouts locate and assess potential targets, evaluating the health, age, and vulnerability of individual prey animals. Once a target is selected, the pack coordinates a complex sequence of behaviors that may include flanking maneuvers, relay chases that exploit different pack members' strengths, and strategic positioning that drives prey toward areas where the terrain favors the hunters. During a typical moose hunt, different wolves assume specialized roles. Lead wolves initiate the chase and test the moose's defenses, while other pack members position themselves to cut off escape routes. Some wolves focus on harassing the moose from behind, nipping at legs and flanks to slow it down, while others attempt to grab the muzzle or throat for a killing bite. This division of labor allows the pack to maintain continuous pressure on prey that might easily escape from a single predator. Lions have developed their own approach to cooperative hunting, one that takes advantage of their social structure and savanna environment. Lionesses, who do most of the hunting, employ sophisticated ambush tactics that require precise timing and coordination. Some members of the pride position themselves as drivers, approaching prey animals from one direction to push them toward concealed hunters waiting in ambush positions. The success of these coordinated hunts depends on complex communication systems that operate across multiple sensory channels. Lions use a combination of vocalizations, body language, and positioning to coordinate their attacks without alerting prey. They have learned to approach from downwind to avoid detection, to use terrain features for concealment, and to time their final rush for maximum effectiveness. Spotted hyenas have perhaps evolved the most sophisticated social hunting system of any terrestrial predator. Their clans can include over 80 individuals operating within complex social hierarchies that determine hunting roles and food sharing arrangements. Large hyena clans can successfully hunt animals as formidable as adult buffalo, using coordinated attacks that exploit the hyena's incredible endurance and bone crushing jaws. Hyena hunting strategies vary depending on prey size and group composition. When hunting smaller antelope, a few individuals may be sufficient, but taking down large ungulates requires the coordination of dozens of hunters. They employ relay chases that can continue for miles, wearing down prey through sustained pursuit rather than explosive speed. Once the prey is exhausted, multiple hyenas attack simultaneously, using their powerful jaws to inflict crippling injuries that prevent escape. Marine environments have given rise to equally sophisticated cooperative hunting strategies. Orcas, the ocean's apex predators, have developed hunting techniques so specialized that different populations employ completely different strategies based on their preferred prey. Some populations have perfected the art of beaching themselves temporarily to catch seals on shore, a behavior that requires not only individual skill but also cultural transmission of knowledge between generations. Other orca populations have developed coordinated hunting strategies for different prey species. When hunting large whales, pods work together to separate calves from their mothers, using their combined mass and intelligence to overcome prey much larger than themselves. When pursuing dolphins or smaller whales, they employ herding behaviors similar to those used by terrestrial pack hunters, surrounding prey and preventing escape. Dolphin societies have independently evolved cooperative hunting strategies that demonstrate remarkable behavioral flexibility. Some populations have learned to drive fish onto beaches where they can be easily caught, while others use coordinated bubble nets to trap schools of fish. Perhaps most remarkably, some dolphin groups use tools in their hunting, cooperating to position marine sponges in ways that help them flush prey from hiding places. The evolution of pack hunting has driven corresponding innovations in prey defense systems. Herd behavior represents the primary defensive response to coordinated predation pressure. When individual prey animals aggregate into groups, they create collective sensory networks that dramatically improve early warning capabilities. A herd of antelope has dozens or hundreds of pairs of eyes, ears, and noses constantly scanning for danger, making it much more difficult for predators to approach undetected. But herding behavior involves more than just collective vigilance. Many prey species have evolved sophisticated alarm systems that allow rapid communication about different types of threats. Meerkats have developed one of the most complex alarm systems known, with different calls for aerial predators, terrestrial threats, and snakes. Each type of call triggers specific defensive responses appropriate to the particular danger. The dilution effect provides another major advantage of group living for prey species. When predators attack a herd, any individual animal's chance of being selected decreases as group size increases. This simple mathematical relationship has driven the evolution of ever larger aggregations in many species. Wildebeest herds on the Serengeti can include hundreds of thousands of individuals, creating a sea of potential targets that makes it difficult for predators to focus on any single animal. Some prey species have developed collective defense strategies that actively repel predators rather than simply trying to avoid detection. Musk oxen form defensive circles when threatened by wolves, presenting a wall of horns and hooves that few predators can penetrate. Adult animals position themselves on the outside of the circle, while calves shelter in the center, creating a fortress-like formation that can withstand sustained attacks. Zebra societies employ a different, but equally effective group defense strategy. When threatened, zebras form tight aggregations where their striped patterns create visual confusion for predators. The mass of moving stripes makes it difficult for hunters to single out individual targets, while the group's collective movement creates a coordinated escape that can overwhelm pursuing predators. Birds have perhaps perfected the art of collective defense through the evolution of flocking behaviors that create some of nature's most spectacular displays. Starling murmurations involve thousands of birds moving in perfect synchronization, creating flowing patterns that can confuse aerial predators while allowing rapid collective response to threats. The mathematical principles underlying these formations ensure that information about predator attacks can propagate through the entire flock within seconds. The social revolution has also transformed the temporal and spatial scales at which predator-prey interactions operate. Cooperative hunting allows predators to sustain longer chases and coordinate attacks across larger areas than would be possible for solitary hunters. This has driven prey species to evolve migration strategies that operate across continental scales, moving between seasonal ranges in patterns that have been refined over millions of years. The Great Migrations of East Africa represent perhaps the most spectacular example of how social behavior has scaled up predator-prey interactions. Millions of wildebeest, zebras, and gazelles move in coordinated patterns across the Serengeti ecosystem, following rainfall and fresh grass growth. This massive movement creates temporary concentrations of prey that support large predator populations, but also spreads grazing pressure across vast areas and reduces the impact of any local predator population. Social learning has become crucial for both predators and prey in these complex systems. Young wolves must learn not only individual hunting skills, but also how to coordinate with pack members, understand social hierarchies, and participate in group decision-making. This cultural transmission of knowledge means that hunting strategies can evolve and improve within a single generation, accelerating the arms race between predators and prey. Prey species similarly rely on social learning to develop effective anti-predator behaviors. Young animals learn to recognize different types of threats, understand alarm calls, and participate in group defense strategies by observing and imitating older, more experienced individuals. This social transmission of knowledge allows prey populations to adapt rapidly to new predator threats or changing environmental conditions. The evolution of social hunting and defense has created feedback loops that have driven the development of increasingly sophisticated cognitive abilities in both predators and prey. Coordinating group behaviors requires advanced communication skills, spatial reasoning, and the ability to understand the intentions and capabilities of both group members and opponents. This cognitive arms race has contributed to the evolution of larger brains and more complex behaviors in many social species. Communication systems have become increasingly elaborate as social complexity has increased. Pack hunting predators must be able to coordinate their actions while remaining silent enough to avoid detection by prey. This has led to the evolution of sophisticated gestural communication systems, subtle vocalizations, and even chemical signaling methods that allow coordination without revealing the pack's presence. Territory and resource defense have added another layer of complexity to social predator-prey systems. Groups that successfully establish and defend territories gain exclusive access to prey resources within their boundaries, but they must also invest considerable energy in territorial defense. This creates complex trade offs between group size, territory quality, and defensive capabilities that influence both predator and prey population dynamics. Seasonal variations in social structure reflect the dynamic nature of these relationships. Many species that live in groups during times of high predation pressure may disperse into smaller units when threats are reduced. These flexible social systems allow animals to optimize their grouping strategies based on current conditions, demonstrating the sophisticated behavioral flexibility that has emerged from millions of years of social evolution. The social revolution in predator-prey relationships has created some of the most complex and dynamic systems in nature. As we observe these intricate social behaviors, we begin to see how they connect to patterns that operate across time itself, where the rhythm of days and seasons creates another dimension of strategic complexity in the eternal dance between hunters and the hunted. Time itself has become a battlefield in the predator-prey arms race. While space provides the arena where encounters unfold, time creates the rhythm that governs when those encounters occur. The strategic use of temporal patterns, daily cycles, seasonal rhythms, and life history timing has allowed species to minimize dangerous overlaps with their enemies or optimize encounters with their prey. In this temporal dimension, survival often depends not on speed or strength, but on precise timing and the ability to predict when opportunity or danger will arrive. The daily cycle of light and darkness creates one of the most fundamental temporal battlegrounds. As the Earth rotates and sunlight sweeps across landscapes, it creates a constantly shifting mosaic of opportunity and vulnerability. Dawn and dusk represent transitional periods when the advantages shift between predators adapted to different light conditions, creating windows of opportunity that many species have learned to exploit with remarkable precision. Owls embody the mastery of nocturnal hunting, their entire anatomy sculpted by millions of years of night hunting. Their enormous eyes gather available light with such efficiency that they can hunt successfully under starlight alone. Specialized feathers create silent flight, while asymmetrical ear openings provide three-dimensional acoustic mapping capabilities that allow them to locate prey by sound alone. These adaptations make them nearly unstoppable predators in the darkness, but they come at a cost. Owls are relatively helpless during bright daylight when their specialized night vision becomes a liability. Their prey have responded by developing complementary temporal strategies. Many small mammals have become primarily diurnal, concentrating their feeding and reproductive activities during daylight hours when owl predation pressure is minimal. Field mice, voles, and shrews that venture out during the day face different predators, hawks, snakes, and weasels, but they have evolved different defensive strategies appropriate to visual hunters rather than acoustic ones. The transition periods at dawn and dusk create particularly complex temporal dynamics. These crepuscular hours represent times when both day and night specialists may be active simultaneously, creating brief windows of intense predation pressure. Many prey species have learned to minimize activity during these dangerous transition periods, retreating to safety during the changing of the predatory guard. Bats and their insect prey have engaged in one of the most sophisticated temporal arms races in nature. As nocturnal aerial predators, bats have driven many flying insects to adopt carefully timed activity patterns that minimize exposure to echolocation-based hunters. Some moths have become day active, abandoning the night sky to their ultrasonic predators. Others have concentrated their activity into brief windows just after sunset or before dawn when bat activity is reduced. The temporal strategies extend beyond simple day night cycles to encompass lunar rhythms that operate on monthly timescales. Many marine organisms time their reproductive activities to specific moon phases, taking advantage of tidal extremes or reduced predation pressure during certain lunar conditions. Grunion fish time their spawning to coincide with the highest tides of the lunar cycle, allowing them to deposit eggs on beaches where aquatic predators cannot reach them. Coral reef communities demonstrate how lunar cycles can create complex temporal patterns of predation and vulnerability. Many reef fish spawn during new moon periods when reduced illumination provides some protection from visual predators. However, this has led to the evolution of specialized predators that concentrate their hunting efforts during these predictable spawning events, creating intense but brief periods of predation pressure. Seasonal timing represents perhaps the most dramatic scale of temporal strategy in predator prey relationships. The annual migration of Arctic terns covers over 40,000 miles as these birds follow summer conditions from Arctic to Antarctic and back again. This extraordinary journey allows them to exploit seasonal peaks in prey abundance while avoiding the harsh winter conditions that would make survival impossible in either polar region. The timing of these migrations requires extraordinary precision. Arriving too early means facing harsh conditions with limited food resources. Arriving too late means missing the peak abundance of prey species. Many migratory species have evolved internal biological clocks that integrate multiple environmental cues, day length, temperature changes, food availability to trigger migration at precisely the right moment. Arctic predators face unique temporal challenges as their environment swings between months of continuous daylight and months of polar night. Polar bears have adapted to these extreme seasonal variations by timing their hunting activities to coincide with optimal ice conditions. They concentrate their feeding during winter and spring when sea ice allows access to seal populations, then fast during summer months when ice retreat forces them onto land. The synchronization of predator and prey life cycles creates temporal bottlenecks where survival depends on precise timing. Many bird species time their nesting to coincide with peak insect abundance, ensuring adequate food supplies for growing chicks. However, climate change is disrupting these carefully calibrated temporal relationships, causing mismatches between predator needs and prey availability that can have catastrophic consequences for population survival. Phenology, the study of seasonal life cycle events, reveals how climate change is creating novel temporal dynamics in predator-prey relationships. Earlier spring warming has advanced the timing of insect emergence in many ecosystems, but the predatory birds that depend on these insects have not always adjusted their migration timing accordingly. This temporal mismatch can result in birds arriving after peak insect abundance has passed, leading to reduced reproductive success and population declines. Some predators have evolved extraordinary flexibility in their temporal strategies, allowing them to exploit multiple prey species with different activity patterns. Coyotes demonstrate remarkable behavioral plasticity, adjusting their hunting schedules based on local prey availability and human activity patterns. In urban environments, they may become largely nocturnal to avoid human interference, while in wilderness areas, they may hunt during daylight hours when it's more energetically efficient. The temporal dimension extends to multi year cycles that can dramatically alter predator prey dynamics. The famous lynx snowshoe hare cycles in Canadian forests operate on roughly 10-year periods, with populations of both species rising and falling in predictable patterns. These cycles demonstrate how temporal patterns can operate across multiple generations, creating boom and bust dynamics that shape entire ecosystems. Insect outbreaks represent another form of temporal strategy where prey species overwhelm predators through synchronized emergence. Periodical cicadas emerge in massive numbers every 13 or 17 years, timing their emergence so precisely that it creates a predator satiation effect. The sheer number of emerging cicadas overwhelms the local predator population's ability to consume them, ensuring that enough individuals survive to maintain the population despite intense predation pressure. These prime numbered cycles may have evolved specifically to avoid synchronization with predator life cycles. By emerging every 13 or 17 years, cicadas minimize the chance that specialized predators could evolve life cycles that would consistently coincide with cicada emergences. This temporal strategy represents one of the most sophisticated examples of evolutionary mathematics in nature. Diapause, a programmed developmental delay, allows many species to synchronize their life cycles with favorable temporal windows while avoiding periods of high predation risk. Many insects time their development so that vulnerable life stages occur during periods when their primary predators are absent or less active. This temporal fine-tuning requires integration of multiple environmental cues and represents a form of biological weather forecasting that can extend months or even years into the future. The evolution of circadian rhythms, internal biological clocks, has provided organisms with the ability to anticipate daily changes in predation, pressure, and prey availability. These internal timekeepers allow animals to prepare for dawn hunting before light levels actually change, or to seek shelter before nocturnal predators become active. The precision of these biological clocks often exceeds that of human timepieces, demonstrating the intense selective pressure for accurate temporal coordination. Some predators have learned to exploit the predictable nature of circadian rhythms in their prey. Certain spiders position themselves in their webs just before the peak activity periods of their preferred prey, optimizing their hunting efficiency by being in the right place at exactly the right time. This temporal precision hunting requires not only accurate biological clocks, but also detailed knowledge of prey behavior patterns. Seasonal fat storage represents another temporal strategy that allows animals to survive periods when the balance between predator and prey shifts unfavorably. Bears accumulate massive fat reserves during salmon runs and berry seasons, then enter hibernation to avoid the energetically expensive winter period when prey is scarce and hunting is difficult. This temporal energy management allows them to effectively skip the season when the predator-prey balance is least favorable. Migration timing has become increasingly critical as climate change alters the seasonal availability of resources and the timing of predator activity. Many species are struggling to adjust their migration schedules to match changing environmental conditions, creating temporal mismatches that can have severe consequences for population survival. The flexibility to adjust timing may become one of the most important factors determining which species can adapt to rapidly changing conditions. Weather patterns create shorter-term temporal opportunities that many predators have learned to exploit. Storms and extreme weather events can disrupt normal prey behavior patterns, creating windows of vulnerability that skilled predators can capitalize on. Some predators actually increase their hunting activity during certain weather conditions when their success rates are higher. The temporal battlefield extends to the molecular level, where circadian clocks control the expression of genes involved in metabolism, sensory perception, and motor activity. These molecular timing systems allow organisms to pre-adapt their physiology for different times of day, optimizing their performance for either hunting or avoiding predation before the critical moment arrives. As we observe these intricate temporal strategies, we see how the dimension of time has become as important as space in shaping predator-prey relationships. The mastery of timing, whether measured in seconds, seasons, or years, often determines success or failure in the eternal dance of survival. These temporal tactics set the stage for understanding how these relationships scale up to engineer entire ecosystems, creating the complex ecological architectures that surround us. The intricate temporal dances between predators and prey ripple outward, shaping not just individual survival, but the very architecture of entire ecosystems. What begins as a simple interaction between hunter and hunted cascades through food webs alters nutrient cycles and can even reshape physical landscapes. These trophic cascades demonstrate how predator-prey relationships function as ecosystem engineers, wielding influence far beyond their immediate participants to create the complex ecological structures that define the natural world. The story of wolves in Yellowstone provides perhaps the most dramatic example of how predators can engineer entire landscapes. When wolves were eliminated from the park in the 1,920 seconds, the ecosystem began a slow transformation that wouldn't be fully understood until decades later. Without predation pressure, elk populations grew rapidly and changed their behavior patterns fundamentally. No longer needing to remain vigilant for wolf packs, elk began spending extended periods in river valleys where the richest vegetation grew. This behavioral shift had profound consequences for plant communities. Aspen, willow, and cottonwood saplings that had previously regenerated successfully in valley bottoms were now subjected to intense, continuous browsing pressure. Young trees were eaten down to stumps year after year, preventing forest regeneration. Over several decades, the absence of wolves led to the collapse of riparian forests throughout much of Yellowstone's northern range. The loss of these forests triggered a cascade of secondary effects that rippled through the entire ecosystem. Without tree cover, stream banks became unstable and began to erode. Water temperatures rose as shade disappeared, affecting fish populations and aquatic invertebrates. Songbird communities that depended on riparian forests declined dramatically. Beaver populations crashed as their preferred food sources vanished, leading to the loss of beaver dams that had created wetland habitats for dozens of other species. When wolves were reintroduced to Yellowstone in 1995, they began to reverse this ecological transformation through what researchers call the landscape of fear. The mere presence of wolves forced elk to alter their behavior patterns, avoiding areas where they might be vulnerable to pack attacks. Valley bottoms, where escape routes were limited and cover was sparse, became dangerous places for elk to linger. This behavioral change allowed vegetation communities to begin recovering. Aspen groves that had been suppressed for decades began producing new shoots. Willows grew taller as browsing pressure decreased, Stream banks stabilized as root systems developed. The returning vegetation created habitat for songbirds, small mammals, and insects that had been largely absent from these areas for generations. The wolf reintroduction demonstrates how predators can influence ecosystems through both direct killing and indirect behavioral effects. While wolves certainly kill elk, their greater impact may come from forcing elk to spend more time in less productive but safer habitats, reducing their overall impact on vulnerable plant communities. This ecology of fear can be as powerful as direct predation in shaping ecosystem structure. Sea otters provide another compelling example of predatory ecosystem engineering, but in marine environments where the mechanisms operate somewhat differently. These charismatic marine mammals are keystone predators in kelp forest ecosystems, maintaining the delicate balance that allows these underwater forests to flourish. Their preferred prey, sea urchins, are voracious grazers capable of creating urchin barrens, underwater deserts, where they have consumed virtually all available algae and kelp. When sea otter populations are healthy, they keep urchin numbers in check through steady predation pressure. This allows kelp forests to develop into complex, three-dimensional habitats that support extraordinary diversity. Giant kelp can grow over a foot per day under optimal conditions, creating underwater canopies that provide shelter, breeding grounds, and feeding areas for hundreds of species. Fish, invertebrates, marine mammals, and seabirds all depend on the complex habitat structure that kelp forests provide. However, when sea otter populations decline due to hunting pressure, disease, or predation by sharks and killer whales, the ecosystem can collapse with surprising speed. Released from predation pressure, urchin populations explode and consume kelp forests faster than they can regenerate. The complex three-dimensional habitat is reduced to flat expanses of rock and sand, supporting only a fraction of the biodiversity that kelp forests maintain. The cascading effects extend beyond the immediate marine environment. Kelp forests are highly productive ecosystems that export significant amounts of organic matter to adjacent habitats. They also provide nursery areas for many commercially important fish species. The loss of kelp forests can therefore affect both nearshore and offshore marine communities, as well as human communities that depend on marine resources. Large predatory fish demonstrate how trophic cascades can operate in open ocean environments. Sharks, tuna, and other apex predators help maintain the structure of marine food webs by controlling populations of mid-level predators. When these apex species are removed through overfishing, the ecosystem can undergo dramatic shifts that affect everything from plankton communities to seabird populations. The decline of large sharks in many ocean regions has led to increases in their prey species, including rays and smaller sharks that feed on shellfish and small fish. These mesopredators can dramatically alter seafloor communities and compete with commercially important species for food resources. Some researchers believe that the collapse of scallop fisheries off the eastern United States may be partially attributed to increased predation pressure from rays whose populations expanded after shark numbers declined. Freshwater ecosystems provide additional examples of predatory ecosystem engineering. The presence or absence of fish in lakes and ponds can determine whether these systems are dominated by clear water and aquatic plants or turbid water and algae. When predatory fish like bass or pike are present, they control populations of planktivorous fish that feed on zooplankton. This allows zooplankton populations to thrive, and these tiny grazers keep algae populations in check, maintaining clear water conditions. Without predatory fish, planktivorous species like minnows and juvenile fish can reach very high densities, reducing zooplankton populations and allowing algae to bloom unchecked. The result is often a turbid green lake with little aquatic vegetation and reduced overall biodiversity. This demonstrates how predator-prey relationships can determine fundamental ecosystem properties like water clarity and primary productivity. Arctic ecosystems provide dramatic examples of how predator effects can cascade across multiple trophic levels and even influence global climate patterns. Arctic foxes, as predators of seabirds, can significantly influence nutrient cycling on islands where large seabird colonies nest. When fox populations are high, they reduce seabird numbers, which decreases the amount of marine-derived nutrients that birds bring to terrestrial ecosystems through their guano. This reduction in nutrient inputs can alter plant community composition and productivity across entire islands. Since many Arctic plants grow slowly and have limited access to nutrients, the loss of seabird-derived fertilizer can cause long-lasting changes in vegetation patterns. These changes in plant communities can then affect other species, from insects to caribou, demonstrating how marine predator-prey relationships can influence terrestrial ecosystems. The role of predators in controlling disease dynamics represents another important aspect of ecosystem engineering. Many predators preferentially target sick, weak, or otherwise compromised individuals, effectively removing potential disease reservoirs from populations. This selective predation can help prevent disease outbreaks that might otherwise devastate prey populations and affect entire communities. Wolves, for example, often target elk infected with chronic wasting disease, a fatal neurological condition that affects cervids. By removing infected individuals, wolves may help slow the spread of this disease through elk populations. Similar patterns have been observed with other predator-prey systems, where predation pressure helps maintain the health of prey populations by removing diseased individuals before they can spread pathogens to healthy animals. The temporal aspects of ecosystem engineering by predators add another layer of complexity to these relationships. Predator effects often operate on multiple timescales simultaneously. Immediate effects include the direct removal of individuals from prey populations. Short-term effects involve behavioral changes in prey species as they adjust their activities to avoid predation. Long-term effects emerge as these behavioral changes allow other species to recover or as genetic changes accumulate in prey populations. Climate change is beginning to disrupt many of these predator-engineered ecosystems by altering the timing and intensity of predator-prey interactions. Warming temperatures are changing the distributions of both predators and prey, creating novel combinations and breaking up evolutionary partnerships that have shaped ecosystems for millennia. Sea ice loss in the Arctic is disrupting predator-prey relationships that depend on stable ice platforms, while changing precipitation patterns are altering the seasonal cycles that many terrestrial predators depend on. The complexity of these ecosystem engineering effects means that predicting the consequences of predator loss or introduction is extremely challenging. Each ecosystem has its own unique web of relationships, and the removal or addition of a predator species can trigger cascades that unfold over years or decades. This unpredictability makes conservation and ecosystem management particularly challenging, as the full consequences of changes may not become apparent until long after intervention opportunities have passed. Human activities have inadvertently conducted large-scale experiments in predator removal and introduction, providing insights into how these ecosystem engineers shape the natural world. The introduction of cats to islands has led to the extinction of countless bird species, while the removal of large predators from many ecosystems has triggered trophic cascades that continue to unfold today. Understanding predator effects as ecosystem engineers is becoming increasingly important as we face global biodiversity loss and ecosystem degradation. Many conservation efforts now focus on maintaining or restoring predator populations, not just for their own sake, but because of their crucial role in maintaining ecosystem structure and function. The wolves of Yellowstone and the sea otters of the Pacific coast have become symbols of how predators can serve as ecosystem architects, shaping the very landscapes they inhabit through their relationships with prey. These ecosystem engineering effects demonstrate that predator-prey relationships extend far beyond simple interactions between individual species. They reveal how these relationships create the complex, interconnected webs that define ecological communities and maintain the diversity of life on Earth. While many predators shape their ecosystems through subtle influences and gradual effects, certain species wield such disproportionate power that their presence or absence can fundamentally alter the character of entire landscapes. These keystone hunters function as ecological cornerstones, supporting the arch of biodiversity that defines their ecosystems. When they disappear, the arch collapses, often with surprising speed and devastating consequences that ripple through every level of the biological community. The concept of keystone species emerged from the pioneering work of ecologist Robert Payne, who conducted a deceptively simple experiment on the rocky shores of Washington state. In 1963, he began removing sea stars from small sections of intertidal habitat, then carefully documented what happened to the communities left behind. The results transformed our understanding of how ecosystems function and revealed the extraordinary power that single species can wield over entire biological communities. The sea star Pisaster ocraceus, though comprising less than 1% of the biomass in its intertidal community, proved to be the keystone that held the entire system together. When Payne removed these predators, the effects cascaded through the community with remarkable speed. Mussels, freed from their primary predator, began to dominate available space on the rocks. Their rapid growth and aggressive space competition quickly crowded out other species that had previously coexisted in the diverse intertidal community. Within just a few years, areas where sea stars had been removed transformed from diverse communities containing over a dozen species to near monocultures dominated by mussels. Barnacles, limpets, chitons, and other invertebrates that had thrived under the sea star's regulatory influence virtually disappeared. The complex three-dimensional habitat structure created by diverse species assemblages flattened into uniform mussel beds that supported far fewer species and ecological relationships. The power of Pisaster lies not in its abundance, but in its feeding preferences and hunting strategy. As a generalist predator with a particular fondness for mussels, it prevents any single prey species from monopolizing limited space resources. By keeping mussel populations in check, the sea star maintains open space where other species can establish and persist. Its selective predation creates a mosaic of different aged mussel patches interspersed with areas available for colonization by other species. This regulatory role demonstrates how keystone predators often function as diversity maintainers rather than diversity reducers. By preventing competitive exclusion, they allow many species to coexist that would otherwise be unable to share the same habitat. The predator's hunting activities create spatial and temporal heterogeneity that increases the number of ecological niches available to other community members. Gray wolves exemplify keystone predators in terrestrial ecosystems, wielding influence that extends far beyond their direct prey relationships. The reintroduction of wolves to Yellowstone revealed how a single predator species can trigger trophic cascades that reshape entire landscapes. Beyond their well documented effects on elk behavior and vegetation recovery, wolves influence dozens of other species through both direct and indirect pathways. The presence of wolves has led to recovery of scavenger communities that had declined during the decades when wolves were absent from the ecosystem. Ravens, eagles, bears, and coyotes all benefit from wolf kills, which provide carrion resources throughout the year. This subsidy effect supports populations of species that might otherwise struggle to find adequate food during harsh winter months. Wolves also compete with and sometimes kill coyotes, leading to changes in small mammal communities. With reduced coyote pressure, populations of rodents and rabbits have increased in some areas, providing more food for hawks, owls, foxes, and weasels. These effects demonstrate how keystone predators can influence species with which they have no direct interaction, creating indirect effects that ripple through multiple trophic levels. The behavioral effects of wolf presence extend to species that are not even potential prey. Scavengers modify their feeding patterns in response to wolf activity, while small carnivores adjust their hunting strategies to avoid areas where wolves are active. Even ravens have been observed changing their foraging behavior in relation to wolf pack movements, demonstrating the pervasive influence of keystone predators on community dynamics. Sea otters represent perhaps the most dramatic example of marine keystone predators, capable of maintaining or destroying some of the ocean's most productive ecosystems. Their role in kelp forest dynamics has been studied extensively, revealing how a relatively small population of marine mammals can control the fate of entire underwater forests. The mechanism of sea otter keystone effects operates through their voracious appetite for sea urchins, invertebrates that can consume kelp faster than it can grow when their populations are unchecked. A single sea otter can consume over 1,000 sea urchins per year, keeping urchin populations at levels that allow kelp forests to flourish. When sea otter populations are healthy, kelp forests can develop into complex vertical structures that support extraordinary biodiversity. The loss of sea otters triggers rapid ecosystem collapse. Without predation pressure, urchin populations explode and create urchin barrens, underwater wastelands, where virtually all macroalgae have been consumed. These barren areas support only a fraction of the species richness found in kelp forests, representing one of the most dramatic ecosystem state changes documented in marine environments. The effects of sea otter presence extend beyond direct kelp forest maintenance. Healthy kelp forests export significant amounts of organic matter to adjacent ecosystems, supporting both nearshore and deep sea communities. They also provide nursery habitat for many commercially important fish species and buffer coastal areas from wave action during storms. The presence or absence of sea otters therefore influences not just marine biodiversity, but also human coastal communities. Jaguar populations in Central and South American rainforests demonstrate how keystone predators can maintain diversity in some of the world's most complex ecosystems. As the largest predators in neotropical forests, jaguars exert top-down control on prey populations that would otherwise dramatically alter forest structure and composition. Jaguars prey on a wide variety of species, including large herbivores like peccaries and deer that can significantly impact forest regeneration through their browsing and foraging activities. By controlling these herbivore populations, jaguars help maintain the complex plant community structure that supports the extraordinary diversity of tropical rainforests. Their hunting activities create a landscape of fear that influences where and when herbivores feed, allowing sensitive plant species to persist in areas where they might otherwise be eliminated by intensive browsing. The cultural and ecological significance of jaguars extends beyond their direct predatory effects. In many indigenous cultures, jaguars are viewed as guardians of the forest, and their presence indicates ecosystem health. This cultural connection has important conservation implications, as traditional knowledge systems often recognize the keystone role of apex predators long before scientific studies document their ecosystem effects. African lions showcase how keystone predators can influence savanna ecosystems through complex interactions with multiple prey species and environmental factors. Lions primarily hunt large ungulates, but their effects extend to smaller species through competitive interactions and habitat modifications created by their hunting activities. The presence of lions influences the behavior and distribution of hyenas, leopards, and cheetahs, creating a complex hierarchy of carnivore interactions that affects prey accessibility for each species. Lions can steal kills from smaller predators and exclude them from prime hunting areas, forcing these species to adjust their hunting strategies and habitat use patterns. These competitive effects cascade down to influence small mammal and bird communities that serve as prey for the smaller carnivores. Lion hunting activities also create opportunities for scavenger communities. A single lion kill can support dozens of species, from large scavengers like hyenas and vultures to small opportunists like insects and birds that feed on scraps. The spatial distribution of lion kills influences nutrient cycling across savanna landscapes, creating patches of enhanced soil fertility that affect plant growth patterns. Polar bears represent a unique case of keystone predators adapted to one of Earth's most extreme environments. As the apex predator of Arctic marine ecosystems, polar bears exert significant influence on seal populations, which in turn affects Arctic marine food webs. Their hunting success depends critically on sea ice extent and timing, making them particularly vulnerable to climate change effects. The loss of polar bears from Arctic ecosystems could trigger cascades that extend far beyond their direct prey relationships. Seals freed from polar bear predation, pressure might increase to levels that affect fish populations and alter Arctic marine productivity. The complex relationships between ice, seals, fish, and polar bears represent one of the most climate-sensitive keystone predator systems on Earth. Tiger sharks demonstrate how keystone predators can maintain diversity in marine ecosystems through their wide-ranging movements and diverse diet. These large sharks consume everything from sea turtles and dolphins to fish and invertebrates, exerting regulatory pressure across multiple trophic levels. Their presence helps prevent any single prey species from dominating marine communities. The loss of tiger sharks and other large sharks from many ocean regions has led to mesopredator release, where medium-sized predators increase dramatically in the absence of apex predation. These mesopredators can then overconsume their preferred prey, leading to cascading effects that alter entire marine food webs. The collapse of some shellfish fisheries has been attributed to increased predation pressure from rays and smaller sharks, whose populations expanded after large shark numbers declined. The conservation of keystone predators presents unique challenges because their effects often extend far beyond protected area boundaries. Wolves, jaguars, and polar bears require vast territories that span multiple jurisdictions and land use types. Their conservation therefore requires landscape scale planning and cooperation between multiple stakeholders with potentially conflicting interests. The economic value of keystone predators often becomes apparent only after they disappear and ecosystem services begin to decline. The cost of losing kelp forests when sea otters disappear includes not just biodiversity loss, but also reduced coastal protection, decreased fish production, and diminished carbon sequestration. Similarly, the loss of wolves can lead to vegetation changes that affect watershed function and recreational values. Climate change poses particular threats to keystone predators because their ecosystem effects often depend on precise timing and environmental conditions. Changes in prey availability, habitat distribution, and seasonal cycles can disrupt the regulatory mechanisms that allow keystone species to maintain ecosystem balance. Understanding and protecting these critical relationships becomes increasingly urgent as environmental change accelerates. The study of keystone hunters reveals the profound interconnectedness of natural systems and the outsized influence that individual species can wield over ecosystem structure and function. As we continue to explore these relationships, we begin to see how they extend into the microscopic realm, where some of nature's smallest predators exert equally dramatic influences on the biological world around us. Beneath the threshold of human perception lies a world where predators and prey engage in struggles as fierce and consequential as any witnessed in the visible realm. In the microscopic universe of bacteria, protists, and viruses, hunters pursue their quarry through landscapes measured in micrometers, wielding molecular weapons and employing strategies that can influence ecosystems on a global scale. These invisible predators may be unseen by the naked eye, but their effects ripple upward through every level of biological organization, shaping the health of individuals, the structure of communities, and even the chemistry of our planet's atmosphere and oceans. Bacteriophages represent perhaps the most abundant predators on Earth, outnumbering their bacterial prey by approximately 10 to 1 in most environments. These viral hunters have evolved exquisite specificity, with each phage species typically capable of infecting only a narrow range of bacterial hosts. Their predatory strategy involves hijacking the cellular machinery of their prey, turning bacteria into factories for producing more viruses before ultimately destroying the host cell in an explosive release of viral progeny. The precision of bacteriophage hunting surpasses that of any macroscopic predator. These viruses have evolved receptor proteins that recognize specific molecules on bacterial cell surfaces with lock and key specificity. Once attached, they inject their genetic material into the host cell, effectively performing a molecular invasion that reprograms the bacterium's entire metabolism. The infected cell ceases its normal functions and instead begins producing viral components, assembling new phage particles with assembly line efficiency. The timing of this molecular predation is precisely controlled by viral genes that monitor resource levels within the host cell. When conditions are optimal, the virus may remain dormant, integrating its DNA into the bacterial chromosome and waiting for the right moment to begin replication. When resources become scarce or the host cell is stressed, the dormant virus activates, rapidly producing hundreds of offspring before lysing the cell to release them into the environment. This microscopic predation has profound effects on bacterial population dynamics and ecosystem function. In marine environments, bacteriophages kill an estimated 20 to 40% of all bacteria daily, effectively controlling bacterial populations and recycling nutrients back into the water column. This viral predation prevents bacterial populations from consuming all available nutrients and helps maintain the delicate balance of marine food webs. Predatory bacteria demonstrate a different approach to microscopic hunting, actively pursuing and consuming other bacterial cells through direct physical interactions. Vibrio bacteriovirus exemplifies this strategy, functioning as a microscopic bacterial wolf that hunts other bacteria with remarkable efficiency. These tiny predators, measuring less than 1 micrometer in length, swim rapidly through their aquatic environments using rotating flagella that propel them at speeds equivalent to a human swimming at several thousand miles per hour. When a vibrio encounters suitable prey, it attaches to the bacterial cell wall and begins boring through the protective outer membrane using specialized enzymes. Once inside the prey's periplasmic space, the predator begins consuming the host cell from within, growing larger as it digests cellular components. Eventually, the elongated predator divides into multiple offspring, which escape by bursting through the weakened cell wall of their consumed host. The specificity of bacterial predation rivals that of much larger hunters. Different vibrio strains show preferences for particular prey species, and their hunting success varies dramatically depending on environmental conditions such as temperature, salinity, and nutrient availability. Some strains have evolved to hunt primarily in soil environments, while others specialize in aquatic habitats, demonstrating ecological specialization comparable to that seen in macroscopic predator communities. Protists represent another major group of microscopic predators, functioning as the primary consumers of bacteria in many aquatic ecosystems. These single-celled eukaryotes employ diverse hunting strategies, ranging from passive filter feeding to active pursuit of prey. Paramecium species use coordinated ciliary action to create water currents that sweep bacteria towards specialized feeding structures, while amoebas actively pursue prey using pseudopodia that can engulf bacterial cells through a process called phagocytosis. The hunting behavior of predatory protists demonstrates remarkable sophistication despite their unicellular organization. Many species can discriminate between different types of prey, preferentially consuming nutritious bacteria while avoiding toxic or unpalatable species. Some protists have evolved the ability to detect chemical signals released by prey bacteria, allowing them to actively track down food sources in their microscopic environment. Tetrahymena species showcase how protist predators can switch between different feeding modes depending on environmental conditions. When bacteria are abundant, they primarily filter feed, using coordinated ciliary action to sweep food particles into their oral grooves. When prey becomes scarce, they can transform into more aggressive hunters, actively pursuing individual bacterial cells and adjusting their swimming patterns to optimize prey encounter rates. The predatory behavior of protists extends to cannibalistic interactions, where larger individuals consume smaller members of their own species. This size-based predation creates complex population dynamics, where growth rates, prey availability, and predation pressure interact to determine community structure. Some protist species have evolved elaborate mating systems that may function partly to reduce cannibalism by allowing individuals to recognize potential mates versus potential prey. Fungal predation represents another fascinating aspect of the invisible hunt, with certain fungi evolving specialized mechanisms for capturing and consuming other microorganisms. Nematode trapping fungi produce elaborate snares and traps that capture microscopic worms with mechanical precision. These hyphal structures function as biological lassos, explosive traps, or adhesive networks that immobilize prey before the fungus begins digestive processes. The oyster mushroom Pleurotus austriatus demonstrates how fungi can function as active predators, producing specialized hyphal structures that pierce nematode cuticles and inject paralyzing toxins. Once immobilized, the nematodes are consumed by fungal hyphae that grow throughout their bodies, converting the prey into fungal biomass. This predatory behavior supplements the mushroom's normal decomposer lifestyle, providing additional nitrogen and other nutrients that may be limiting in its environment. Carnivorous plants blur the boundaries between the microscopic and macroscopic worlds of predation, using sophisticated trapping mechanisms to capture prey ranging from bacteria to small insects. The aquatic bladderwort Utricularia generates negative pressure within specialized bladder traps that can suck in prey in less than a millisecond. These traps are so sensitive that they can be triggered by bacteria brushing against trigger hairs, demonstrating predatory behavior that operates across multiple size scales simultaneously. The digestive processes employed by carnivorous plants involve complex communities of microorganisms that break down captured prey. Within the digestive chambers of pitcher plants, specialized bacterial communities produce enzymes that decompose insect prey, while protists consume bacteria and small organic particles. This creates miniature ecosystems within individual plants, where microscopic predator-prey relationships play out in fluid-filled chambers. Parasitic organisms represent a unique form of microscopic predation, where the hunt extends over much longer timescales than typical predator-prey interactions. Malaria parasites demonstrate extraordinary sophistication in their hunting strategies, manipulating multiple host species to complete their complex life cycles. Within mosquito hosts, the parasites alter feeding behavior to increase transmission rates, while in human hosts, they hide from immune system detection by constantly changing their surface proteins. The molecular arms race between parasites and host immune systems represents one of the most intense selection pressures in biology. Parasites must constantly evolve new strategies to evade immune detection, while hosts develop increasingly sophisticated defense mechanisms to recognize and eliminate invaders. This microscopic warfare drives rapid evolution in both parasites and hosts, contributing to the extraordinary genetic diversity observed in immune system genes. Viral manipulation of host behavior extends to effects on macroscopic organisms, demonstrating how microscopic predators can influence the visible world. The rabies virus alters brain function in infected mammals, increasing aggressive behavior and promoting transmission through biting. Toxoplasma gondii manipulates the behavior of infected rodents, making them less fearful of cats and more likely to be consumed by the parasite's definitive host. The ecological effects of microscopic predation extend far beyond individual host-parasite relationships. In aquatic ecosystems, viral predation on bacteria and algae influences primary productivity, nutrient cycling, and the flow of carbon through food webs. Marine viruses play crucial roles in controlling algal blooms and maintaining diversity in phytoplankton communities by preventing any single species from dominating available resources. Soil ecosystems harbor some of the most complex microscopic predator-prey relationships on Earth. The soil food web includes multiple levels of microbial predators, from bacterial predators that consume other bacteria to predatory protists that regulate bacterial populations. These microscopic interactions control nutrient cycling processes that determine plant growth and ecosystem productivity in terrestrial environments. Climate change is beginning to alter microscopic predator-prey relationships in ways that could have significant ecosystem-level consequences. Rising temperatures affect the metabolic rates of microorganisms differently, potentially disrupting established predator-prey ratios. Ocean acidification may alter the effectiveness of viral predation in marine environments, while changes in precipitation patterns could affect soil, microbial communities, and their associated predator-prey dynamics. The study of microscopic predation has practical applications for human health and environmental management. Understanding viral predation of bacteria has led to renewed interest in phage therapy as an alternative to antibiotics for treating bacterial infections. Knowledge of protist predation is being applied to biological water treatment systems, while research on fungal predators may lead to new biological control agents for agricultural pests. Antibiotic resistance in bacteria creates new dynamics in microscopic predator-prey relationships. Some research suggests that predatory bacteria and viruses may be more effective at controlling antibiotic-resistant pathogens than traditional chemical treatments. This has led to investigations into using natural microbial predators as therapeutic agents, essentially harnessing evolved predatory relationships for medical applications. The invisible hunt reveals that predator-prey relationships operate at every scale of biological organization, from molecular interactions within cells to global biogeochemical cycles. These microscopic hunters may be unseen, but their influence extends throughout the visible world, shaping the health of organisms, the structure of communities, and the functioning of entire ecosystems. As we begin to understand these hidden relationships, we see how the principles governing macroscopic predation extend seamlessly into the molecular realm, creating a unified understanding of how life shapes and is shaped by the eternal dance between hunters and their prey. Water transforms the fundamental physics of predation, creating a three-dimensional battlefield where the rules governing terrestrial hunting no longer apply. In aquatic environments, predators and prey navigate a medium 800 times denser than air, where sound travels 4 times faster, light penetrates only limited distances, and the very properties of fluid dynamics reshape every aspect of the hunt, from the microscopic plankton drifting in surface waters to the massive predators prowling the deepest ocean trenches. Aquatic ecosystems have given rise to predatory strategies and defensive adaptations found nowhere else on Earth. The physics of water movement creates unique opportunities for both hunters and their prey. Unlike terrestrial environments where friction with solid surfaces dominates locomotion, Aquatic animals must master the complex fluid dynamics of swimming through a three-dimensional medium. This fundamental difference has driven the evolution of body forms and propulsion systems that represent some of the most elegant engineering solutions in the natural world. Sharks exemplify the mastery of aquatic predation, their body design refined over 400 million years of evolutionary pressure. The classic shark form, streamlined, torpedo-shaped, with precisely positioned fins, represents a near-perfect solution to the engineering challenges of underwater hunting. Their heterocircle tail generates both forward thrust and lift, while paired fins provide stability and maneuverability. The slight negative buoyancy of most sharks allows them to sink rapidly when not actively swimming, an adaptation that aids in ambush tactics and energy conservation. The great white shark demonstrates how aquatic predators can exploit the three-dimensional nature of water to achieve devastating surprise attacks. Their hunting strategy involves approaching prey from below, using the darkness of deep water as camouflage while their white bellies blend with the bright surface when viewed from above. This countershading allows them to approach within striking distance before launching vertical attacks that can propel their massive bodies completely out of the water. The mechanics of these breaching attacks reveal the extraordinary power that aquatic predators can generate. Great whites must accelerate from depths of 30 feet or more to achieve the speed necessary to launch themselves airborne while carrying prey that may weigh several hundred pounds. This requires burst speeds approaching 25 miles per hour and precise timing to intersect with fast-moving seals at the ocean's surface. Barracuda employ a different aquatic hunting strategy, relying on sustained high-speed pursuit rather than ambush tactics. Their elongated body form minimizes drag, while powerful tail muscles generate the thrust needed for extended chases. Unlike the explosive power of sharks, barracuda hunting depends on endurance and the ability to maintain high speeds over considerable distances through open water. The hydrodynamics of high-speed aquatic pursuit create unique challenges for both predators and prey. At high velocities, water resistance increases exponentially, requiring enormous energy expenditure to maintain pursuit speeds. This has led to the evolution of specialized adaptations like the rigid body form of tuna, which reduces energy loss through body flexion, and the precisely shaped fins that maintain laminar flow around the swimming body. Electric predation represents one of the most sophisticated hunting strategies to evolve in aquatic environments. Electric eels generate voltages up to 600 volts, creating electrical fields powerful enough to stun fish at distances of several feet. This bioelectric hunting system allows them to disable prey without physical contact, effectively extending their strike range through the conductive properties of water. The precision of electric hunting systems rivals the most advanced human technology. Electric rays can modulate their discharge intensity based on prey size and proximity, delivering just enough voltage to stun without wasting energy on excessive electrical output. Sharks and other cartilaginous fish have evolved electroreceptors so sensitive they can detect the electrical fields generated by the muscle contractions of hidden prey, essentially allowing them to hunt by reading the bioelectric signatures of their targets. Ambush predation reaches extraordinary sophistication in aquatic environments where the three-dimensional nature of water allows predators to attack from any direction. Anglerfish demonstrate the ultimate in aquatic ambush strategy, using bioluminescent lures to attract prey in the perpetual darkness of deep ocean waters. Their hunting method requires no active pursuit, Instead, they have evolved into living traps that exploit the visual systems of their prey. The frogfish takes aquatic ambush hunting to artistic extremes, combining perfect camouflage with explosive strike mechanisms. These masters of disguise can alter their coloration and skin texture to match their surroundings so precisely that they become virtually indistinguishable from coral, sponges, or rocky surfaces. When prey approaches within range, they can extend their jaws and engulf prey in a suction feeding attack that occurs in as little as 6 milliseconds. Suction feeding represents a uniquely aquatic hunting mechanism that has no terrestrial equivalent. By rapidly expanding their buccal cavity, aquatic predators create powerful inward water flows that draw prey into their mouths along with the surrounding water. This feeding strategy allows relatively small predators to capture prey much larger than their mouth size would suggest possible, as the prey is essentially hydraulically transported into the predator's digestive system. The moray eel has evolved perhaps the most sophisticated variation of suction feeding through its pharyngeal jaw system. While its primary jaws capture prey through conventional biting, a second set of jaws in its throat can extend forward to grasp captured prey and transport it deeper into the digestive system. This adaptation allows morays to consume prey much larger than their head size and helps explain their success as ambush predators in coral reef environments. Cooperative hunting in aquatic environments demonstrates how water's three-dimensional nature can be exploited by social predators. Dolphin pods employ sophisticated herding techniques that take advantage of water's fluid properties to concentrate and trap prey. By swimming in coordinated formations, they can create pressure waves and turbulence that confuse schools of fish and prevent their escape. Some dolphin populations have developed even more sophisticated cooperative hunting strategies, such as mud plume feeding, where they use their tail flukes to stir up sediment that creates temporary barriers around fish schools. Others employ bubble net feeding, releasing streams of bubbles that create cylindrical nets around prey. These techniques demonstrate how intelligent predators can manipulate the physical properties of water to create tools for hunting. Orcas represent the pinnacle of cooperative aquatic hunting, with different populations developing specialized techniques for different prey species. Some populations have learned to create waves that wash seals off ice floes, while others use coordinated attacks to separate whale calves from their mothers. The cultural transmission of these hunting techniques means that orca populations in different regions have developed distinct hunting traditions that are passed down through generations. The vertical nature of aquatic environments creates unique opportunities for predation across different depth zones. Deal vertical migration, where vast numbers of organisms move between deep and shallow waters on daily cycles, represents one of the largest animal migrations on Earth. Predators throughout the water column time their activities to intercept these migrating masses of prey. Sperm whales exemplify deep water hunting specialization, diving to depths of over 3,000 feet to hunt giant squid in the perpetual darkness of the deep ocean. These hunts may last over an hour, requiring extraordinary physiological adaptations for breath holding and pressure tolerance. The battles between sperm whales and giant squid represent some of the most epic predator-prey encounters in the natural world, though they occur in depths where human observation is nearly impossible. The bioluminescence of deep-sea predators creates a unique sensory environment where light itself becomes a weapon. Many deep-sea predators use photophores to create complex light displays that can confuse, attract, or communicate with other organisms. The vampire squid can eject bioluminescent mucus when threatened, creating a glowing cloud that may confuse predators long enough for escape. Filter feeding represents a massive scale approach to aquatic predation, where the largest animals on Earth capture some of the smallest prey. Baleen whales use specialized feeding structures to strain vast quantities of krill and small fish from the water, essentially functioning as living plankton nets. The efficiency of this feeding strategy allows blue whales to sustain their enormous body size on prey that individually weigh less than a gram. The seasonal aggregations of filter feeding whales create some of the most spectacular predatory events in the oceans. When humpback whales gather in feeding areas, they may employ cooperative bubble net feeding techniques that concentrate prey into dense patches before engulfment. These feeding events can involve dozens of whales working together to maximize capture efficiency. Parasitic and symbiotic relationships in aquatic environments often blur the lines between predation and mutualism. Cleaner fish stations on coral reefs represent complex ecological relationships where small fish remove parasites from larger species, gaining nutrition while providing a cleaning service. However, some predators have evolved to mimic cleaner fish, using this deception to approach close enough to bite chunks of flesh from unsuspecting hosts. The remora's relationship with sharks demonstrates how aquatic environments enable unique forms of commensalism that would be impossible on land. By using specialized suction discs to attach to large marine predators, remoras gain transportation and access to food scraps, while apparently providing little benefit to their hosts. This relationship allows small fish to exploit the hunting success of large predators without the energy costs of independent swimming. Ocean currents add another dimension to aquatic predation by creating highways that transport both predators and prey across vast distances. Many marine predators time their movements to coincide with current patterns that concentrate prey organisms. Tuna and other pelagic predators follow thermal fronts and upwelling areas where nutrient-rich waters support high prey densities. The seasonal patterns of current-driven productivity create temporal hotspots where predators from across entire ocean basins may converge. The California current system supports massive aggregations of seabirds, marine mammals, and fish during upwelling seasons when deep, nutrient-rich waters support explosive phytoplankton growth and the food webs it supports. Freshwater environments present their own unique challenges and opportunities for aquatic predation. The lower salinity of freshwater affects buoyancy and osmoregulation, while the typically smaller scale of freshwater systems creates different predator-prey dynamics than those found in marine environments. Many freshwater predators must be generalists, adapting to the more variable and often more seasonal nature of freshwater prey communities. Pike and other freshwater ambush predators demonstrate how aquatic hunting strategies adapt to the structural complexity of lakes and rivers. Their elongated body form and powerful burst swimming ability make them effective hunters in the vegetated shallows, where many freshwater prey species seek refuge. The clear water of many freshwater systems also places greater emphasis on visual hunting than is typical in marine environments. As we observe these diverse aquatic hunting strategies, we see how water has served as an evolutionary laboratory for developing predatory innovations found nowhere else in nature. The three-dimensional battlefield of aquatic environments continues to shape predator-prey relationships that span from surface waters to the deepest ocean trenches, creating some of the most spectacular and sophisticated hunting strategies on our planet. The dimension of height adds complexity to predator-prey relationships that transforms simple ground-level encounters into elaborate three-dimensional chess games played across multiple vertical zones. From the forest floor to the canopy tops, from shallow coral gardens to towering reef walls, vertical ecosystems create distinct layers of habitat that support specialized predator-prey communities at each level. These stratified environments function as apartment buildings of biodiversity, where different species occupy specific floors, and the movement between levels becomes as important as any horizontal migration across landscapes. Tropical rainforests represent perhaps the most complex vertical predator-prey systems on Earth, with distinct ecological zones stacked from ground level to heights exceeding 200 feet. Each layer supports its own community of predators and prey, many of which rarely venture beyond their preferred vertical habitat. This stratification has driven the evolution of specialized hunting strategies adapted to the unique conditions found at different heights within the forest structure. The forest floor exists in perpetual twilight, where less than 2% of sunlight penetrates the dense canopy overhead. In this dim environment, predators rely heavily on non-visual senses to locate prey. Army ants demonstrate the ultimate in ground-level cooperative hunting, forming massive columns that sweep across the forest floor like living carpets of destruction. Their raids can include over 1 million individual ants, moving in coordinated waves that flush out and overwhelm ground-dwelling invertebrates. The three-dimensional structure of army ant raids exploits the vertical complexity of forest floor habitats. While the main column advances across the ground, smaller groups climb into low vegetation and fallen logs, ensuring that prey cannot escape simply by climbing a few feet above the forest floor. This multi-level hunting strategy allows army ants to capture prey throughout the entire vertical range of the ground zone habitat. Jaguar hunting strategies demonstrate how large predators navigate the vertical complexity of rainforest environments. These powerful cats are equally comfortable stalking prey on the ground, climbing into trees to hunt arboreal mammals, or even entering water to catch fish and caimans. Their muscular build and powerful claws allow them to carry prey weighing over 100 pounds up into trees, effectively removing their kills from potential scavengers and competitors that cannot follow them into the canopy. The understory layer, extending from about 6 to 60 feet above ground, creates a twilight zone where dappled sunlight creates a complex mosaic of light and shadow. This vertical habitat supports its own specialized predator community adapted to hunting in the filtered light and dense vegetation of the middle forest layers. Many understory predators are ambush specialists, taking advantage of the visual complexity created by tangled vines, dense foliage, and shifting patterns of light and shadow. Fer-de-lance vipers exemplify understory ambush predation, using their cryptic coloration and patient hunting style to remain motionless for weeks while waiting for suitable prey to pass within striking distance. Their vertical positioning on low branches allows them to intercept both terrestrial and arboreal prey, effectively hunting in two different vertical zones simultaneously. Heat-sensing pit organs allow them to detect warm-blooded prey in the dim understory environment, where visual hunting would be ineffective. Birds of prey demonstrate remarkable vertical specialization within forest ecosystems. Different hawk species partition the vertical habitat by hunting at different heights and using different flight patterns adapted to specific forest layers. Sharp-shinned hawks specialize in high-speed pursuit through dense understory vegetation, their short wings and long tails providing exceptional maneuverability in cluttered environments. In contrast, broad-winged hawks hunt from the canopy, using their broader wings to soar above the forest and spot prey in clearings below. The forest canopy, beginning around 60 feet above ground and extending to heights of 200 feet or more in tropical forests, represents the most species-rich layer of these vertical ecosystems. Here, the majority of forest animals live their entire lives without ever touching the ground, moving through an aerial highway system of branches, vines, and epiphyte gardens that can support the weight of everything from tiny insects to large primates. Canopy predators have evolved remarkable adaptations for hunting in three-dimensional aerial environments. Many have developed prehensile tails that function as fifth limbs, allowing them to maintain secure grips while pursuing prey through the swaying branches. Others have evolved gliding membranes that allow them to move between trees without descending to lower forest levels where different predators might threaten them. Harpy eagles represent the apex predators of neotropical forest canopies, using their extraordinary agility and powerful talons to hunt sloths, monkeys, and other arboreal mammals. Their hunting strategy involves patient observation from concealed perches, followed by explosive flights through dense vegetation that require split-second navigation around branches and vines. The power of their strike is so great that they can snatch adult sloths from branches and carry them through the canopy to feeding perches. Spider communities demonstrate remarkable vertical partitioning within forest ecosystems, with different species constructing webs at specific heights and using hunting strategies adapted to the prey available at their chosen level. Ground-dwelling wolf spiders actively hunt prey on the forest floor, while orb weavers construct aerial nets at various heights to intercept flying insects. Some spider species migrate vertically on a daily basis, hunting near the ground at night and retreating to higher levels during the day. The emergent layer, consisting of the tallest trees that extend above the main canopy, creates yet another distinct vertical habitat zone. These forest giants experience conditions more similar to open savanna than to the forest interior, with intense sunlight, strong winds, and extreme temperature fluctuations. The predators that hunt in this aerial zone must be adapted to these harsh conditions while maintaining the ability to navigate the complex three-dimensional structure of the tree crowns. Coral reefs create equally complex vertical predator-prey systems in marine environments, with distinct communities occupying different depth zones, from the shallow reef flats to the deep reef walls that plunge into blue water. Each vertical zone supports specialized predators adapted to the specific conditions of light, current, and prey availability found at different depths. The shallow reef flats, bathed in bright sunlight and swept by strong currents, support visual predators that rely on speed and agility to capture prey in the high-energy environment near the surface. Reef sharks patrol these areas using their superior swimming ability to chase down fast-moving fish, while giant trevally use their powerful build and explosive acceleration to overwhelm schools of smaller fish with devastating charge attacks. As depth increases and light levels diminish, the predator community shifts towards species adapted for hunting in darker conditions. Many deeper reef predators are nocturnal, remaining hidden in caves and crevices during the day and emerging at night when their superior night vision gives them an advantage over prey species adapted to daylight conditions. Moray eels exemplify this strategy, using their excellent night vision and sensitive chemical detection to hunt sleeping fish that are vulnerable in the darkness. The reef wall represents a unique vertical habitat where predators must adapt to hunting on steep surfaces while dealing with strong currents that can sweep both predators and prey away from the reef structure. Many wall-dwelling predators are ambush specialists that use the complex topography of the reef to conceal themselves while waiting for prey to venture within striking distance. Groupers demonstrate how large reef predators exploit the vertical structure of coral reefs. These powerful fish often hunt by positioning themselves near the bottom of walls or in sandy channels between reef structures, using their explosive acceleration to intercept prey moving between different reef zones. Their hunting success depends on precise timing and positioning that takes advantage of the three-dimensional complexity of reef environments. The drop-off zone, where coral reefs plunge into deep blue water, creates a unique interface between reef and pelagic environments. Predators hunting in this zone must be adapted to both the structured reef environment and the open water conditions beyond the reef edge. Many species that hunt in this zone are vertical migrators, moving up and down the reef wall to exploit prey that migrate between deep and shallow waters on daily cycles. Kelp forests represent another form of vertical marine ecosystem, with giant kelp creating underwater forests that can reach heights of over 100 feet. These submarine forests support complex predator-prey communities at different levels, from bottom-dwelling predators that hunt among the holdfasts to surface-oriented species that patrol the kelp canopy where light levels are highest. Sea otters hunting in kelp forests demonstrate remarkable three-dimensional navigation skills as they dive to depths of over 100 feet to hunt sea urchins and other bottom-dwelling prey. Their hunting success depends on their ability to navigate through the complex kelp forest structure while holding their breath for extended periods. Upon capturing prey, they must return to the surface through the same complex three-dimensional maze. Mountain ecosystems create vertical gradients where predator-prey communities change dramatically with elevation. Different species occupy distinct altitudinal zones, and the movement of prey species between these zones creates opportunities for predators adapted to hunt at specific elevations. Many mountain predators are vertical migrants themselves, following prey species as they move to different elevations seasonally. Snow leopards demonstrate supreme adaptation to vertical hunting in mountain environments, using their powerful build and exceptional agility to pursue prey across steep, rocky terrain where other large predators cannot follow. Their hunting strategy often involves traversing vast vertical distances to locate and stalk prey, sometimes hunting across elevation changes of several thousand feet during a single pursuit. Urban environments create novel vertical predator-prey systems as cities develop increasingly complex three-dimensional structures. Peregrine falcons have adapted to hunting in urban canyons, using skyscrapers as substitute cliff faces and exploiting the abundant pigeon populations that thrive in cities. Their hunting strategy involves high-speed dives from building tops that can exceed 200 miles per hour in the confined spaces between tall buildings. The seasonal changes in vertical ecosystems add temporal complexity to these already intricate spatial relationships. Many vertical habitats experience dramatic seasonal changes in structure as deciduous forests lose their leaves and expose previously hidden predator-prey interactions to new visual and physical conditions. Snow cover can eliminate lower vertical zones entirely, forcing both predators and prey to concentrate in higher levels where food and shelter remain available. Climate change is beginning to alter the vertical structure of many ecosystems, potentially disrupting the specialized predator-prey relationships that have evolved to exploit specific vertical zones. Rising temperatures are causing upward shifts in the altitudinal ranges of many mountain species, while changing precipitation patterns are altering the vertical structure of forests and other layered ecosystems. As we observe these complex vertical relationships, we see how the dimension of height has created some of the most diverse and specialized predator-prey communities on Earth, each adapted to the unique conditions found at their preferred level in these living skyscrapers of biodiversity. The annual cycle of seasons creates a temporal kaleidoscope that continuously reshapes predator-prey relationships, transforming abundance into scarcity, altering landscapes, and driving some of the most spectacular movements of life on Earth. As temperature, daylight, and precipitation patterns shift throughout the year, both hunters and hunted must adapt their strategies to survive periods of plenty and endure times of hardship. These seasonal rhythms have become so deeply embedded in the biology of species that they influence everything from reproduction timing to migration patterns, creating intricate annual choreographies that have been refined over millions of years. Winter represents the ultimate test of survival strategies in temperate and polar regions, where plummeting temperatures and snow cover can transform productive ecosystems into apparent wastelands. For predators, winter presents the dual challenge of reduced prey availability and increased energy demands for thermoregulation. Many species have evolved remarkable adaptations that allow them to maintain their hunting effectiveness even under the most challenging conditions. Snowy owls exemplify predatory success in winter environments, their entire biology tuned to exploit the brief abundance that occurs during peak rodent population years in the Arctic tundra. These powerful hunters time their reproduction to coincide with lemming population explosions, which follow roughly 4-year cycles driven by complex interactions between vegetation, herbivores, and predators. During peak lemming years, a single pair of snowy owls may raise up to 10 offspring, taking advantage of prey densities that can exceed 1,000 lemmings per hectare. The hunting strategy of snowy owls during winter demonstrates remarkable adaptation to seasonal conditions. Their exceptional hearing allows them to locate prey moving beneath snow cover, while their heavily feathered feet provide insulation and traction on icy surfaces. Unlike most owl species that are strictly nocturnal, snowy owls hunt during the continuous daylight of Arctic summers and adapt to winter conditions by becoming active during the brief daylight hours when prey movement is most predictable. Arctic foxes showcase another approach to winter predation, employing both active hunting and scavenging strategies that shift based on seasonal prey availability. During summer, they primarily hunt small mammals, birds, and their eggs, taking advantage of the brief period when Arctic ecosystems burst with reproductive activity. As winter approaches and prey becomes scarce, Arctic foxes increasingly rely on cached food stores and carrion, often following polar bears to scavenge seal carcasses. The seasonal coat change of Arctic foxes represents one of nature's most dramatic adaptations to changing predation pressures. Their brown summer fur provides camouflage against the tundra landscape, while their thick white winter coat offers both insulation and concealment against snow-covered terrain. This seasonal transformation requires enormous metabolic investment, but provides crucial advantages for both hunting success and predator avoidance throughout the annual cycle. Winter dormancy strategies demonstrate how some species essentially opt out of the seasonal predator-prey game during the most challenging periods. Bears enter hibernation not because they cannot find food in winter, but because the energy cost of active hunting in harsh conditions exceeds the potential benefits. Their ability to survive months without eating depends on massive fat accumulation during autumn when salmon runs and berry crops provide concentrated energy sources that can sustain them through the dormant period. The timing of hibernation entry and emergence has profound effects on predator-prey dynamics in temperate ecosystems. Bears that emerge early from hibernation may find limited food availability and face the challenge of feeding growing cubs when prey species are just beginning their own reproductive cycles. Climate change is beginning to disrupt these carefully timed relationships, with earlier spring warming causing mismatches between bear emergence and peak prey availability. Spring represents a season of opportunity and vulnerability as ecosystems emerge from winter dormancy, and both predators and prey face the challenges of reproduction and territorial establishment. The timing of spring activities often determines reproductive success for the entire year, creating intense selection pressure for precise phenological timing that maximizes opportunities while minimizing risks. Migratory predators demonstrate some of the most spectacular seasonal strategies, timing their movements to exploit temporary concentrations of prey that occur during specific seasons. Peregrine falcons breeding in the Arctic must time their reproduction to coincide with the brief period when shorebird chicks provide abundant food for growing falcon nestlings. The margin for error is extremely narrow. Arriving too early means facing harsh conditions with limited food, while arriving too late means missing the peak abundance of vulnerable prey. The Great Migrations of the Serengeti exemplify how seasonal strategies operate at ecosystem scales, with millions of herbivores following predictable patterns that support entire communities of predators. Lions, leopards, cheetahs, and hyenas all adjust their territorial behaviors and hunting strategies to exploit the seasonal movements of wildebeest, zebras, and gazelles. Some predator populations follow the herds, while others remain in territories that provide year-round resources but experience dramatic seasonal fluctuations in prey density. Seasonal breeding strategies create temporal windows of vulnerability that predators have learned to exploit with remarkable precision. Many bird species time their nesting to coincide with peak insect abundance, but this also creates predictable opportunities for nest predators. Snakes, raccoons, and other egg predators often show peak activity during the breeding seasons of their prey species, demonstrating how seasonal strategies can cascade through multiple trophic levels. The emergence of periodic cicadas represents one of the most extraordinary seasonal strategies in nature, with some species emerging in massive numbers every 13 or 17 years. These prime-numbered cycles may have evolved specifically to minimize overlap with predator life cycles, ensuring that specialized predators cannot evolve to consistently exploit cicada emergences. The overwhelming abundance of emerging cicadas creates a predator satiation effect, where even intensive predation cannot significantly impact the overall population. Autumn presents unique opportunities and challenges as many species prepare for winter, while others take advantage of seasonal resource pulses like fruit crops and salmon runs. The timing of these autumn events can determine winter survival for many predator species, creating intense competition for access to the most productive hunting areas during critical preparation periods. Salmon runs create some of the most spectacular seasonal predation events on Earth, concentrating enormous biomass in small areas for brief periods. Brown bears, black bears, wolves, eagles, and numerous other predators converge on salmon streams during peak runs, creating temporary communities of predators that would rarely interact under normal circumstances. The hierarchical access to prime fishing spots often reflects complex social dynamics and size based dominance relationships among the assembled predators. The cultural transmission of seasonal hunting strategies adds complexity to these relationships, particularly among long lived social species. Orca populations have developed distinct seasonal hunting traditions that are passed down through generations, with different pods specializing in different prey species during specific seasons. Some populations focus on salmon runs during summer months, while others specialize in marine mammal hunting that peaks during different seasonal periods. Seasonal fat storage represents a crucial adaptation for predators that must survive periods of food scarcity. Many species show remarkable physiological flexibility, building fat reserves during periods of abundance that can sustain them through lean times. The efficiency of this energy storage often determines survival success during harsh seasons, creating strong selection pressure for optimal foraging strategies during productive periods. The seasonal dynamics of predator prey relationships extend to marine environments where ocean productivity cycles drive complex temporal patterns of abundance and distribution. Upwelling seasons create temporary hotspots of productivity that support massive aggregations of seabirds, marine mammals, and fish. The timing of these productive periods varies geographically, creating seasonal migration corridors that predators follow across entire ocean basins. Seasonal ice formation in polar regions creates dramatic changes in predator prey accessibility. Polar bears depend on sea ice formation to access their primary prey, seals, while the timing and extent of ice formation determines hunting success for the entire year. Climate change is dramatically altering these ice cycles, forcing polar bears to adapt to shorter hunting seasons and longer periods of food scarcity. The phenological mismatches created by climate change represent one of the most significant threats to seasonal predator prey relationships. Earlier spring warming has advanced the timing of insect emergence in many ecosystems, but migratory bird predators have not always adjusted their arrival timing accordingly. These temporal disconnects can result in predators arriving after peak prey abundance has passed, leading to reduced reproductive success and population declines. Seasonal strategies in tropical environments often revolve around precipitation patterns rather than temperature cycles, with dry and wet seasons creating dramatic changes in habitat structure and prey availability. Many tropical predators show seasonal changes in hunting behavior and territorial patterns that reflect the shifting distribution of water and prey resources throughout the annual cycle. The seasonal regeneration of vegetation creates predictable patterns of herbivore abundance that predators have learned to exploit. Many carnivores time their own reproductive cycles to coincide with the birth of herbivore young, taking advantage of inexperienced prey during vulnerable early life stages. This synchronization requires precise timing that often depends on environmental cues that can be disrupted by changing climate patterns. Desert ecosystems demonstrate extreme seasonal strategies, where brief rainy periods trigger explosive reproduction in prey species that must complete their entire life cycles during short windows of favorable conditions. Predators in these environments often show remarkable behavioral flexibility, switching between active hunting during productive periods and energy conservation during extended droughts. As we observe these intricate seasonal strategies, we see how the annual cycle of environmental change has shaped some of the most spectacular adaptations in predator prey relationships, creating temporal rhythms that connect species across continents and drive some of the most dramatic movements of life on our planet. The rise of human cities has created entirely novel ecosystems where predator prey relationships must adapt to artificial landscapes unlike anything that existed during millions of years of evolutionary history. These urban environments present both unprecedented challenges and unexpected opportunities, forcing wildlife to navigate a world of glass towers, concrete canyons, and artificial lighting while exploiting new food sources and habitat niches that exist nowhere in nature. The speed at which some species have adapted to urban life reveals the remarkable behavioral plasticity that allows predator prey relationships to persist even in the most transformed landscapes. Urban environments fundamentally alter the sensory landscape that predators and prey depend on for communication, navigation, and detection. The constant noise pollution from traffic, construction, and human activity masks many of the acoustic signals that species use to locate prey or detect approaching predators. Light pollution eliminates the natural darkness that many nocturnal hunters require for effective predation, while the vertical glass surfaces of buildings create visual confusion that can disorient both hunters and their prey. Peregrine falcons have perhaps made the most spectacular adaptation to urban hunting, transforming from cliff-dwelling predators into downtown residents that nest on skyscrapers and hunt in urban canyons. Their adaptation to city life demonstrates how behavioral flexibility can allow specialized predators to exploit novel habitat configurations. The vertical architecture of cities provides abundant nesting sites that closely mimic their natural cliff habitats, while the concentrations of pigeons and other urban birds create prey densities far higher than those typically found in natural environments. The hunting behavior of urban peregrines reveals remarkable adaptations to the unique physics of city landscapes. Their high-speed hunting dives must now navigate between buildings, around antennas, and through the complex wind patterns created by urban architecture. These challenges have led to subtle changes in hunting techniques, with urban peregrines often using building updrafts to gain altitude more efficiently and timing their attacks to exploit the channeling effects of city streets. Red-tailed hawks have similarly adapted to urban hunting, shifting from their typical preference for open country to become successful predators in city parks, green corridors, and even dense urban neighborhoods. Their diet in urban areas often differs significantly from their rural counterparts, with increased consumption of rats, pigeons, and other urban adapted prey species. Some urban hawks have learned to hunt at night under streetlights, behavior rarely observed in rural populations. The success of urban raptors has created cascading effects throughout urban ecosystems. Reduced pigeon populations in areas with established raptor territories have allowed other bird species to exploit urban habitats with less competition. The presence of hawks has also influenced the behavior of urban mammals, with squirrels and rats showing increased vigilance and altered activity patterns in areas where raptors hunt regularly. Mammalian predators face greater challenges adapting to urban environments due to their larger space requirements and more conspicuous presence. However, some species have shown remarkable adaptability to city life. Coyotes have become established in cities across North America, demonstrating extraordinary behavioral plasticity as they learn to exploit urban food sources while avoiding dangerous encounters with humans and vehicles. Urban coyotes have developed hunting strategies specifically adapted to city environments. They have learned to use storm drains and greenway corridors for travel and have adjusted their activity patterns to avoid peak human activity periods. Their diet in urban areas often includes a higher proportion of anthropogenic food sources, from garbage to pet food, while they continue to hunt traditional prey like rodents and rabbits in urban parks and vacant lots. The social structure of urban coyote populations often differs from their rural counterparts, with many urban coyotes living as mated pairs or solitary individuals rather than forming the larger packs common in rural areas. This behavioral adjustment may reflect both the reduced territory sizes available in urban environments and the different prey base that supports smaller social groups. Foxes have shown similar adaptability to urban life, with red fox populations becoming established in cities worldwide. Urban foxes often display bolder behavior than their rural counterparts, adapting to the constant presence of humans and learning to exploit human associated food sources. Their hunting behavior in cities combines traditional predation on small mammals with opportunistic scavenging of human refuse and pet food. The temporal patterns of urban predation often differ dramatically from those in natural environments. Many urban predators have shifted their activity patterns to avoid peak human activity periods, becoming more nocturnal or crepuscular than their rural counterparts. This temporal partitioning allows them to exploit urban resources while minimizing dangerous interactions with humans and vehicles. Street lighting has created novel opportunities for some predators while disadvantaging others. Insectivorous bats in some urban areas have learned to exploit the concentrations of insects attracted to streetlights, effectively using human infrastructure to concentrate their prey. However, light pollution also disrupts the circadian rhythms of many species and can interfere with the navigation systems that nocturnal predators depend on for successful hunting. Urban water features, from stormwater retention ponds to ornamental fountains, have created novel aquatic habitats that support unique predator-prey communities. Great blue herons and other wading birds have adapted to hunting in urban water bodies, exploiting populations of introduced fish and amphibians. These artificial aquatic systems often support higher prey densities than natural water bodies due to reduced competition and the absence of natural predators. The introduction of exotic species into urban environments has created novel predator-prey relationships that have no natural counterparts. House cats, perhaps the most successful urban predator, kill billions of birds and small mammals annually in cities worldwide. Their hunting pressure represents an entirely anthropogenic predation source that native prey species have no evolutionary experience with, often leading to severe impacts on urban wildlife populations. Domestic cats demonstrate hunting behaviors that differ from those of wild felids, primarily in their lack of dependence on hunting success for survival. Well-fed house cats continue to hunt actively, often killing prey they do not consume. This surplus killing behavior creates predation pressure that extends beyond what prey populations might experience from wild predators with similar population densities. The vertical structure of urban environments has created novel three-dimensional hunting opportunities that some predators have learned to exploit. Window strikes represent a form of inadvertent predation where building surfaces kill millions of birds annually. Some urban predators have learned to exploit these casualties, with cats and other scavengers regularly patrolling areas beneath tall buildings where bird strikes are common. Urban environments also support thriving populations of synanthropic species that have co-evolved with human settlements. Rats and mice represent prey species that have specifically adapted to urban environments, developing behavioral and physiological traits that allow them to exploit human-created resources. Their success creates opportunities for urban predators, but also presents unique challenges due to their association with human disease transmission and property damage. The management of urban predator-prey relationships requires balancing conservation goals with human safety and property protection concerns. Wildlife management in urban areas often focuses on maintaining viable predator populations while minimizing conflicts with human residents. This can involve habitat modification, population monitoring, and public education programs designed to promote coexistence. Urban habitat fragmentation creates edge effects that can intensify predation pressure on some species while providing refuge for others. Small urban parks and green spaces often support higher predator densities than comparable areas in continuous natural habitats due to the concentration effect of limited habitat availability. These edge effects can create ecological traps where prey species are attracted to apparently suitable habitat that actually exposes them to increased predation risk. The artificial lighting of urban environments has disrupted natural predator-prey cycles in complex ways. Some nocturnal predators have difficulty hunting under bright artificial lighting, while others have adapted to exploit the changed behavior of prey species disoriented by light pollution. Migrating birds that become disoriented by city lights often fall prey to urban predators that have learned to exploit these predictable concentrations of vulnerable individuals. Climate change interactions with urbanization create additional complexity for urban predator-prey relationships. Urban heat island effects can extend the active seasons of both predators and prey, while extreme weather events may concentrate wildlife in urban refugia where different predation dynamics emerge. The role of urban environments as climate refugia is becoming increasingly important as extreme weather events become more frequent. The study of urban predator-prey relationships provides insights into the adaptability of natural systems and the potential for wildlife conservation in human-dominated landscapes. Urban environments serve as natural experiments in rapid adaptation, revealing which species and relationships can persist under extreme habitat modification and which require more natural conditions to maintain viable populations. As urbanization continues to expand globally, understanding how predator-prey relationships adapt to city life becomes increasingly important for maintaining biodiversity in landscapes dominated by human infrastructure. The success stories of urban wildlife adaptation provide hope for conservation in the Anthropocene, while the failures highlight the importance of maintaining natural habitat connectivity and minimizing the impacts of urban development on wildlife populations. The study of predator-prey relationships stands on the threshold of revolutionary discoveries as new technologies provide unprecedented insights into the hidden lives of hunters and their quarry. Advanced tracking systems, molecular techniques, and artificial intelligence are revealing aspects of these ancient relationships that were completely invisible to previous generations of researchers. These technological frontiers promise to transform our understanding of how predators and prey interact across scales, from molecules to entire ecosystems, while also revealing how rapidly changing environments are creating novel evolutionary pressures that may reshape these relationships in the decades to come. Satellite telemetry and GPS tracking have already begun to reveal the extraordinary complexity of predator movements and hunting strategies that were previously impossible to document. Arctic wolves, equipped with high-resolution GPS collars, have shown that their hunting territories can span thousands of square miles, with pack movements coordinated across vast distances through mechanisms that researchers are only beginning to understand. The precision of modern tracking technology allows scientists to document individual hunting events, measure pursuit distances, and analyze the decision-making processes that determine when predators commit to attacks. Real-time tracking of both predators and prey simultaneously is providing new insights into the spatial and temporal dynamics of predator-prey encounters. Studies that track wolves and their elk prey in real time reveal that successful hunts often depend on complex spatial positioning that unfolds over hours or even days, with wolves using landscape features and elk behavior patterns to optimize their hunting success. These detailed movement data are revealing that predation is far more strategic and less opportunistic than previously assumed. The miniaturization of tracking technology is extending these capabilities to smaller species that were previously impossible to monitor continuously. Hummingbirds weighing less than 3 grams now carry transmitters that can track their movements with meter-scale precision, revealing how these tiny predators navigate complex three-dimensional environments while hunting insects. Similar advances are allowing researchers to track the movements of small mammals, reptiles, and even large insects, opening new windows into predator-prey relationships across the entire size spectrum of animal life. Environmental DNA, eDNA, technology represents another revolutionary tool that is transforming how researchers study predator-prey interactions. By sampling water or soil and analyzing the genetic material present, scientists can now determine which species are present in an environment and even identify predator-prey relationships through the detection of prey DNA in predator fecal samples. This technique allows for non-invasive monitoring of predator diets and can detect rare or cryptic species that might never be observed through traditional survey methods. Metabarcoding of predator stomach contents and fecal samples is revealing diet complexity that was previously impossible to document. Traditional diet studies relied on identifying partially digested remains, which often provided incomplete pictures of predator feeding behavior. Modern molecular techniques can identify prey species from tiny DNA fragments, revealing that many predators consume far more diverse diets than previously known, and that their feeding behavior varies dramatically across seasons, habitats, and environmental conditions. Stable isotope analysis provides complementary information about predator-prey relationships by tracking the flow of nutrients through food webs over extended time periods. Different prey species often have distinct isotopic signatures based on their diets and habitats, and these signatures are preserved in predator tissues. This allows researchers to reconstruct feeding relationships and estimate the relative importance of different prey species to predator nutrition, even when direct observation of feeding behavior is impossible. Advanced camera trap technology is providing unprecedented insights into predator behavior and hunting success rates. Modern camera traps can operate continuously for months, capturing thousands of images that document predator prey encounters in detail. Machine learning algorithms are now being developed to automatically analyze these vast image datasets, identifying species, classifying behaviors, and measuring hunting success rates across entire predator communities. Thermal imaging and infrared camera systems are revealing nocturnal predator prey interactions that have remained largely hidden from human observation. These technologies show how body temperature differences between predators and prey create detection opportunities and reveal thermoregulatory strategies that influence hunting behavior. Some researchers are even developing systems that can track individual animals through forest environments using thermal signatures, providing continuous monitoring of predator prey encounters in complex habitats. Acoustic monitoring systems are opening new frontiers in understanding the communication aspects of predator prey relationships. Automated recording systems can monitor soundscapes continuously, detecting predator vocalizations, prey alarm calls, and the acoustic signatures of predator prey encounters. Machine learning algorithms trained to recognize specific sounds can process months of recordings to identify patterns in predator prey interactions that would be impossible to detect through traditional observation methods. Bioacoustic analysis is revealing that many predator prey interactions involve complex acoustic strategies previously unknown to science. Some predators produce infrasonic calls below the range of human hearing to coordinate hunting behavior, while others use acoustic mimicry to deceive prey species. Prey species are being discovered to have far more sophisticated alarm call systems than previously recognized, with different calls providing specific information about predator type, distance, and threat level. The integration of multiple sensor types into comprehensive monitoring systems is creating unprecedented datasets that capture predator-prey relationships in their full complexity. Research sites equipped with networks of camera traps, acoustic monitors, and environmental sensors can now document ecosystem-level predator-prey dynamics continuously, revealing patterns that emerge only through long-term, high-resolution monitoring. Artificial intelligence and machine learning are becoming essential tools for analyzing the massive data sets generated by these new monitoring technologies. Deep learning algorithms can now identify animal species from camera trap images with accuracy exceeding that of human experts, while neural networks trained on movement data can predict predator hunting success based on GPS tracks alone. These AI systems are beginning to reveal patterns in predator-prey behavior that are too subtle or complex for human researchers to detect directly. Predictive modeling using machine learning is enabling researchers to forecast how predator-prey relationships might respond to environmental changes. By training models on historical data that includes both predator-prey dynamics and environmental variables, scientists can begin to predict how climate change, habitat modification, and other environmental changes might affect these crucial ecological relationships. Virtual reality and advanced visualization technologies are providing new ways to understand and communicate the three-dimensional complexity of predator-prey environments. Researchers can now create immersive virtual reconstructions of hunting environments that allow them to analyze spatial relationships and movement patterns from perspectives impossible in the real world. These tools are particularly valuable for understanding complex three-dimensional environments like forest canopies and coral reefs. Biomechanical analysis using high-speed video and force measurement systems is revealing the physics of predator-prey interactions in unprecedented detail. Ultra-high-speed cameras can capture the mechanics of snake strikes, bird stoops, and fish attacks at temporal resolutions that reveal previously unknown aspects of predatory behavior. Force sensors embedded in artificial prey can measure the power of predator attacks and the effectiveness of different hunting strategies. Physiological monitoring of free-ranging animals is providing insights into the energetic costs and benefits of different predator-prey strategies. Heart rate monitors, accelerometers, and other physiological sensors can now track the energy expenditure of hunting behavior in real time, revealing how predators balance energy costs against hunting success rates. These studies are showing that optimal hunting strategies often involve complex trade-offs between energy expenditure, success probability, and prey quality. Genomic analysis is revealing the evolutionary history of predator-prey relationships and identifying the genetic basis of hunting and defensive adaptations. Whole genome sequencing of predators and their prey can identify genes under selection pressure due to predator-prey interactions, revealing how these relationships drive evolution at the molecular level. Comparative genomics across related species is showing how predator-prey arms races have shaped the evolution of entire gene families. CRISPR and other gene editing technologies are beginning to allow experimental manipulation of predator-prey relationships in controlled settings. Researchers can now modify specific genes involved in predatory behavior or prey defense mechanisms to test hypotheses about the genetic basis of these interactions. While these techniques raise important ethical considerations, they offer unprecedented opportunities to understand the molecular mechanisms underlying predator-prey relationships. Climate change is creating natural experiments that are revealing how rapidly predator-prey relationships can adapt to changing environmental conditions. Long-term datasets from research sites around the world are documenting shifts in predator-prey dynamics as temperatures rise, precipitation patterns change, and extreme weather events become more frequent. These studies are revealing both the resilience and vulnerability of these relationships to environmental change. The development of early warning systems for ecosystem collapse is becoming increasingly important as environmental change accelerates. Researchers are working to identify indicators of predator-prey relationship breakdown that could serve as early warning signals for ecosystem-level problems. These systems might allow for intervention before predator-prey relationships collapse entirely, potentially preventing ecosystem-level cascades. Restoration ecology is beginning to incorporate sophisticated understanding of predator-prey relationships into ecosystem restoration projects. Rather than simply reintroducing individual species, restoration programs are increasingly focused on rebuilding functional predator-prey networks that can maintain themselves over time. This approach requires detailed understanding of how these relationships function and how they can be successfully reestablished in degraded environments. Conservation genetics is revealing how small population sizes and habitat fragmentation affect the evolutionary potential of predator-prey relationships. Genetic monitoring of predators and prey in fragmented landscapes is showing how reduced genetic diversity can limit the ability of these species to adapt to changing conditions or co-evolve in response to each other. Urban ecology research is revealing how predator-prey relationships adapt to novel environments created by human activities. Studies of urban wildlife are showing that some predator-prey relationships can persist and even thrive in cities, while others are incompatible with urban environments. This research is informing efforts to design urban landscapes that can support wildlife while meeting human needs. The future of predator prey research will likely be increasingly interdisciplinary, integrating insights from ecology, evolution, physiology, behavior, genetics, and technology. As the tools available to researchers become more sophisticated and the data sets they generate become larger and more complex, new patterns and relationships will undoubtedly emerge that will transform our understanding of these fundamental ecological interactions. The urgency of understanding predator prey relationships is increasing as human activities continue to alter ecosystems around the world. The insights gained from current research frontiers may be crucial for maintaining the ecological relationships that have shaped life on Earth for hundreds of millions of years and ensuring their persistence in an uncertain future. As our journey through the world of predators and prey draws to a close, we find ourselves standing at the edge of a vast understanding that stretches back through deep time to the very origins of complex life on Earth. The relationships we have explored, from the molecular precision of viral hunters to the ecosystem engineering power of apex predators, represent threads in a tapestry so intricate and ancient that it predates the existence of every major lineage of life we recognize today. This eternal dance between hunters and hunted has not merely shaped individual species, it has fundamentally sculpted the architecture of life itself. The predator prey relationship emerges as perhaps the most fundamental organizing principle in biology, a force as constant and pervasive as gravity in shaping the evolution of living systems. Every organism alive today, including ourselves, exists as the product of countless predator prey interactions that have filtered, refined, and directed evolutionary change across billions of years. The genes we carry, the behaviors we exhibit, and even the ecosystems that surround us all bear the indelible signatures of this ancient struggle between hunters and their quarry. Consider the profound implications of what we have discovered in our exploration: the chemistry of neurons, the architecture of eyes, the mechanics of flight, the social structures of communities, the timing of biological clocks, and the geographic distribution of species across the planet. All have been shaped by the evolutionary pressures created when one organism attempts to consume another. The predator prey interaction functions as a universal selection pressure that has influenced virtually every aspect of biological form and function. The arms races we have examined reveal evolution not as a gradual, steady process, but as a dynamic interplay of innovation and counter innovation that can drive rapid change when selection pressures intensify. The molecular precision of cone snail toxins, the acoustic sophistication of bat moth interactions, and the visual deception of camouflage systems all represent solutions to evolutionary challenges posed by the hunt. These solutions often involve such exquisite refinement that they surpass human engineering capabilities, demonstrating the extraordinary creative power of evolutionary processes when driven by the life and death stakes of predation. The temporal dimension of predator-prey relationships reveals how these interactions operate across every conceivable timescale, from the millisecond strike of a mantis shrimp to the million-year evolutionary arms races that have shaped entire lineages. The seasonal migrations that span continents, the circadian rhythms that coordinate daily activities, and the multi-generational cycles that synchronize population dynamics all demonstrate how time itself has become a strategic element in the eternal dance between predators and prey. The spatial complexity we have explored shows how predator-prey relationships have filled every available niche on Earth, from the deepest ocean trenches to the highest mountain peaks, from the microscopic spaces between soil particles to the vast expanses of open ocean. The three-dimensional nature of aquatic hunting, the vertical stratification of forest communities, and the global patterns of migration all reveal how these relationships have come to define the very structure of life on our planet. Perhaps most remarkably, we have seen how predator-prey relationships function as ecosystem engineers, wielding influence that extends far beyond the immediate participants to shape entire landscapes and determine the distribution of countless other species. The wolves of Yellowstone, the sea otters of kelp forests, and the sharks of coral reefs all demonstrate how individual predator-prey relationships can serve as keystone interactions that hold together complex ecological communities. The microscopic world has revealed that these principles operate with equal importance at scales invisible to human perception. The viral predation that controls bacterial populations, the protist hunters that regulate microbial communities, and the parasitic manipulations that alter host behavior all show how predator-prey relationships create the foundation for all higher-level ecological processes. The invisible hunt occurring in every drop of water and grain of soil supports the visible world we experience directly. As we have traced these relationships through urban environments and examined their responses to climate change, we begin to understand both their resilience and their vulnerability. Some predator-prey relationships have shown remarkable adaptability, with species rapidly adjusting their behaviors and strategies to exploit novel environments and changed conditions. Others have proven brittle, collapsing when environmental changes disrupt the delicate timing or spatial relationships that millions of years of evolution have refined. The technological frontiers we have explored promise to reveal even deeper layers of complexity in these relationships. As our ability to track, monitor, and analyze predator-prey interactions becomes more sophisticated, we will undoubtedly discover new aspects of these ancient relationships that will further expand our understanding of how life operates and evolves. The integration of molecular biology, artificial intelligence, and advanced tracking technologies is already revealing patterns and mechanisms that were completely unknown just decades ago. Yet perhaps the most profound insight from our exploration is the recognition that humans have become a novel force in predator-prey relationships, often disrupting patterns that have persisted for millions of years while simultaneously creating entirely new ecological dynamics. Our species has achieved a unique position as a predator capable of hunting virtually any species on Earth, while also serving as an inadvertent prey species for microscopic pathogens and parasites. We have become ecosystem engineers on a global scale, with our activities influencing predator-prey relationships in every biome on the planet. This realization brings with it both great responsibility and unprecedented opportunity. Our growing understanding of how predator-prey relationships function provides us with the knowledge needed to make more informed decisions about conservation, restoration, and sustainable resource management. We now know that protecting apex predators can restore entire ecosystems, that maintaining migration corridors can preserve evolutionary processes, and that managing predator-prey relationships is essential for maintaining the ecological services that human societies depend on. The future of predator-prey relationships on Earth will largely be determined by human choices made in the coming decades. Climate change, habitat destruction, pollution, and species introductions all threaten to disrupt predator-prey relationships that have shaped life for hundreds of millions of years. Yet our increasing ability to understand and predict these relationships also gives us the power to intervene constructively, potentially preserving these crucial ecological interactions even as environments continue to change. The study of predator-prey relationships also offers profound insights into our own nature as biological organisms embedded within ecological systems. We are simultaneously predators and prey, hunters and hunted, shaped by the same evolutionary forces that have molded every other species on Earth. Our intelligence, our social structures, our technologies, and even our cultural institutions all bear the imprint of our evolutionary history as both hunters and potential prey. As we look toward the future, the eternal dance between predators and prey will undoubtedly continue, but it will do so in a world increasingly influenced by human activities. The challenge before us is to ensure that this dance can persist, maintaining the ecological relationships that have created and sustained the diversity of life we have explored. This will require not just scientific understanding, but also wisdom, humility, and a deep appreciation for the intricate beauty of the natural world. The predator-prey relationship represents one of the most ancient and fundamental aspects of life on Earth, a relationship so basic and pervasive that it has become woven into the very fabric of biological existence. From the first cellular predators that consumed their neighbors in primordial seas, to the complex multi-species hunting coalitions that roam modern landscapes, this relationship has been the driving force behind much of evolution's creativity and complexity. In the quiet moments before dawn, that spider still waits in her web while the fly buzzes nearby, unaware of the silk trap that could end its journey. This simple scene, replicated billions of times across the planet every day, connects us to the deepest patterns of life on Earth. In understanding these relationships, we come to understand something fundamental about the nature of life itself, its creativity, its complexity, its resilience, and its profound interconnectedness. The eternal dance continues, and in its endless variations, we glimpse the extraordinary beauty and complexity of the living world that surrounds and sustains us all.